So hey students, welcome to Vedantu NEET English. This is Basavaraj, I'm your biology master teacher. So how is everyone doing? Let me know in the chat right now because I am doing amazing. Because I'm thrilled because this is one of those class plant. Good evening each and every one of you. So students, hope you're able to see me. Students, if you're able to see me, if my voice is visible, audible to you, if my presentation is visible to you, thumbs up in the chat, fire in the chat, hearts in the chat, lightning in the chat, and we will start today's from the basics, from the very basics. What is the meaning of photosynthesis? All the way from photosynthesis. We will go to the advanced topics. I will teach you today cyclic, non-cyclic photosynthesis, phosphorylation, what is C3 cycle, what is C4 cycle, what is chemiosmotic hypothesis, what are the different factors involved in photosynthesis. So each and every step by step I will spoon feed you. I have questions for you, all the NEET PYQs for you. So today's session, students, today's session is the only session you will need for understanding plant physiology. So can we start? Good evening each and every one of you. Divya, Crazy, hello, Ravindrani, each and every one of you. Hello, good evening each and every one of you. So can we start the class? Can we start the amazing class today, that is photosynthesis in higher plants? This is personally my favorite chapter because every single concept, right? Every single concept is so interrelated here, so interrelated. So while we wait for everyone to join, I'll give you 2 minutes. I'll give 2 minutes for everyone to join. I have a simple question. So students, you know, in my class, before I start my class, I ask you a simple question all the time. Today's question is, what is the full form of PCR with respect to? What is PCR with respect to photosynthesis? What is PCR cycle? What is PCR cycle with respect to photosynthesis? Anyone in the chat who can tell me? Anyone in the chat who can tell me what is a full form of PCR cycle with respect to photosynthesis? Everyone in the chat who can tell me? While we wait, I want to answer to this question. Simple question. What is PCR cycle? Anyone in the chat who can tell me? Gayatri, Ravindrani, Lakshmita, Shreyas, Amulya, Crazy, Gaurav, right? Who can tell me? No, it is not polymerized chain reaction. So students, this is nothing but your photosynthetic carbon reduction cycle. Photosynthesis, photosynthate carbon reduction cycle. The other name for this PCR cycle is C3 cycle, also known as your Calvin cycle, right? Also known as Calvin cycle. That is PCR cycle with respect to photosynthesis. Photosynthetic carbon reduction cycle. Can we start? Can we start the class today with an amazing information? So before I start, everyone, like the video. I have been noticing you are not liking my videos. I'm like, why? Why is no one liking the videos? Do you not like the sessions? So students, everyone, like the video, subscribe to the channel, and please share this video as much as possible. Bring your friends, because trust me students, today I'll be teaching you from very basic, the most basic, to the advanced topic. Okay? So this is a Telegram channel. You can join the Telegram channel. The notes for today's class will be available on this Telegram channel. You can also join my Telegram channel where I'll be providing you all the doubts and everything. And we are following the Super 6 series. All of you know this. That is, I did plant life cycles, done and dusted. All of you know the plant cycles now. Gymnosperm, pteridophytes, algae, bryophytes, done and dusted. Then your organism environment, day for yesterday, your Gopika ma'am did it. Then inductive effect, yesterday the Shreyas Sir, amazing class. Even I was attending the class. That is rotational mechanism, biggest monster you slayed. But today we start the most difficult, most demanding chapter from photosynthesis, from plant physiology, that is photosynthesis in higher plants. After today's class, you all of you can say that I am done. nothing can, no question I can solve. Every single question is easy for me. You will have that confidence of photosynthesis. Then tomorrow we have resonance, then we have ray optics, and finally on this Saturday we'll be doing chikungunya and dengue. Okay, let me give you a quick overlook of today's class. We'll be starting with photosynthesis. What is the meaning of photosynthesis? What is the basic definition of photosynthesis? Then we'll be doing requirements of photosynthesis. If you want to perform a photosynthesis, not you or me, we can't do photosynthesis. Who will do? Plants. If plants has to do photosynthesis, what are the requirements? Then we'll be doing some early experiments. Yes, we'll be showing you some experiments which you might have done in your previous classes, in your class 10th, right? In your class 11th. Then we'll be doing photosynthetic pigments and spectrum. What is the difference between action spectrum and your absorption spectrum? The basic difference I'll be telling you. Then we'll be finally be doing light reactions. What are the different photosystems? PS1, what is PS2? Do not worry students, if you have zero knowledge, you do not know anything, you do not even know what is pigment, I will teach you today what is pigment. Don't worry, pigment is not just color. I will teach you what is the meaning of pigment in botany. Then we'll be doing chemiosmotic hypothesis. Then we'll be doing your dark reactions, which is also called as light independent reaction. Then photorespiration, C4 cycle, factors affecting, and finally in the end I will be teaching you a summary. I'll give you a proper summary of the entire chapter. So can we start? Can we start the class today? You know the drill now. All of you like the video and we will start today's amazing class, that is photosynthesis. Photosynthesis in higher plants. I want to see fire, lightning, hearts in the chat, and we will start today's class. Can we start? So all of you know the basic outcome now. So I will teach you the most basic concept today first. What is the meaning of photosynthesis? Basic word. All of us know. Students, write down with me. All of you know, forget photosynthesis for now. All of you know plants are said to be autotrophs. Right? Plants are said to be autotrophs. What is the meaning of autotrophs? The meaning of autotrophs is that plants can prepare their own food. Yes, plants can prepare their own food. What about heterotrophs? Such as you, me, all of us, we are heterotrophs. All the humans, all the animals are heterotrophs. Yes, we are heterotrophs in nature. What is the meaning of heterotrophs? We derive the nutrition. Students, I'm taking the basic level, the most basic level today. We are heterotrophs. We directly or indirectly depend on plants. Yes, we will directly eat a plant. If you are vegetarian, how many of you are vegetarian in the chat? Tell me in the chat, who can tell me how many of you are vegetarians? And some of them are non-vegetarians, which are carnivores, which eat herbivores. Yes, so that are heterotrophs. So I told you autotrophs can prepare their own food. The process, the basic process of preparing the own food, it's called as photosynthesis. It's called as what? Photosynthesis. Now this is a class 6th definition. All of you can agree with me? All of you can agree with me, sir, this is a class 6 definition. But right now, I'm in class 11th, I'm in class 12th, I'm in class 13. What is class 13? I'm a NEET dropper, sir. I need a high level definition. All of you will need? Yes. But students, before I give you the high level definition, understand the basics. What happens in photosynthesis? Understand the basics. What is the meaning of photo? Photo means what? Photo means light. Photo means light. Synthesis means to produce. Yes, photo means light. Synthesis means to produce something. We are preparing a food. Not we, plants. Plants are preparing food using light. Yes, sir, that is correct. But sir, where does this photosynthesis happen? All of us know photosynthesis happen in the green plants. Yes, sir. Photosynthesis happens in the green plant. Which part? Can anyone in the chat tell me which part of the plant? All of us know it happens in the leaf, or some cases herbaceous stem also. True. On the leaf, we have a special cells. The special cells are called as mesophyll cells. How many of you heard it? How many of you know what is mesophyll cells? Inside this mesophyll cells, students, inside the mesophyll cells, we have a structure called as chloroplast. We have a structure called as what? Chloroplast. Chloroplast. Inside the chloroplast, I repeat again, students, basic botany. Inside the chloroplast, the photosynthesis takes place. With me, all of you till now? Is every student out there with me till now? I will ask you, students, if all of you with me, I will continue next. Any doubt, I take up. Okay? Till here, everything is clear? Yes, sir, it is clear. Now, let's look at inside a cell now. Inside a chloroplast. I told you, every single plant out there, not only me, your teachers from school, they have told you, plants take up CO2. All of you know that. Basic botany. Yes, sir. Plants require CO2. Yes, plants are taking up CO2. Inside the chloroplast, inside the plant cell, the mesophyll cell, CO2 is getting converted to a type of sugar, right? Like your glucose, right? Simple sugar like your glucose. Now, can I tell you one line? All of you should agree with me. If you do not agree, you can ask me. From inside your cell, your CO2 is getting converted. Listen to me. Listen to my words. Inside the cell, CO2 is getting converted to glucose. Yes, sir. It is getting converted. When it is getting converted, can you notice one difference here? That is, there is addition of hydrogen. Yes, sir. There is addition of hydrogen. With me, all of you? Basic. Yes, sir. There is addition of hydrogen. In chemistry, addition of hydrogen is called as reduction. Yes. What is oxidation then, sir? Oxidation is loss of electrons or a loss of hydrogen. But here we are gaining hydrogen. So can I call photosynthesis a reduction process? Yes, sir. I can call photosynthesis a reduction process. We are getting advanced now. We are getting advanced. It is a reduction process. With me, all of you? With me? It is a reduction process. First point clear? The second point, students. It requires the hydrogen. Where do you think the hydrogen comes from? Simple botany, simple general knowledge. The hydrogen comes from all the rain, water which you add to your plants. This hydrogen comes from the water. This hydrogen right here comes from the water. All of you add to your dear plants. Even I have a garden. How many of you have a garden in the house? Because I have an amazing garden where I have all the different type of plants. C3 plants, C4 plants, every single plant. Now when this hydrogen is sent there, what happens is oxygen? Oxygen is released outside. Oxygen released outside. And that is the reason we tell. That is the reason we tell plants take up CO2 and they release oxygen outside. Yes, sir, they take up the hydrogen. The splitting of water is happening here. The water is being split up. That we'll come to later, but I'm teaching you basic right now. Water is being split up here and oxygen is released outside. Hydrogen is sent here. Yes, sir, agree with me till here? All of you agree with me till here? Next point. Write down with me all of these points. The next point which I want all of you to focus here is, in photosynthesis, you have learnt in your younger classes that we require sunlight. Plants need sunlight. Yes, sir, plants will need sunlight. So plants are absorbing the sunlight. Yes, agree with me? Plants are absorbing the sunlight. So can I call it a endothermic reaction? They're absorbing the heat. Yes, sir, we can call this an endothermic reaction. Your Photorespiration, photosynthesis is a endothermic reaction. There's reduction is happening, and what is the first point here I told you? CO2, right? CO2 is reduced to, can I tell CO2 is reduced to your C6H12O6. C6H12O6. Till here, all of you are with me? All of you are with me till now? The last point about photosynthesis, which I want all of you to embed in your head. In photosynthesis, plants convert, listen to me very carefully. In photosynthesis, plants convert inorganic substances to organic substances. All of you agree with me? In photosynthesis, plants convert inorganic substances to organic substances. Agree with me? Yes, sir, I agree with you. So can I call it a anabolic process? Can I call it anabolic process? Students, I want you to understand this. I can increase the speed, but I don't want to right now. I want all of you to understand from basics. This is a very easy chapter. Very easy chapter if you understand these words. So can I call it an anabolic process? Yes, sir, I can call it a anabolic process. Anabolic process. Now students, from here onwards, so many NEET PYQs have actually come. So many NEET PYQs have just come from this much of information. Just about photosynthesis, and that's all. This is photosynthesis for you students. All of you know what is photosynthesis now? So I'll give you a definition of photosynthesis now. Photosynthesis is a physico-chemical process. There is a physical thing happening, that is exchange of gases is happening. Yes sir, physical chemical. What is chemical? There is a chemical reaction taking place here. So it's a physico-chemical process. What is photosynthesis? Photosynthesis is a physico-chemical process in which carbon dioxide is reduced to glucose. Water is broken down, and that water is broken down where hydrogen is given to your glucose and oxygen is liberated out. Here, water is broken down. This is your photosynthesis. This is photosynthesis. Clear? Till now, the basic thing you understood? Clear, all of you? It's a physical-chemical process where carbon dioxide, water reacts in the presence of sunlight, endothermic, and this I'll get to it. This I will get. I'll tell you what is pigment. I'll tell you what is chlorophyll. Convert to glucose plus water. And light energy is converted to chemical energy. As Shreyas Sir would have told you, in physics, energy cannot be created. In physics, you might have learned energy cannot be created nor be destroyed. It can be transferred from one form to another form. Yes, sir. All of you will agree with Shreyas Sir here. Energy can be only transferred. So, can I tell you, pigment is that substance. Pigment is a chemical compound. Pigment is a chemical compound which facilitates, which converts light energy to your chemical energy. From light energy to chemical energy's conversion is done with the help of chlorophyll, which is a pigment. What is it? It is a pigment. So what is a pigment, students? I'll get to this later in detail later on. I will tell you in general right now. All of you know, when you hear pigment, you think of color. Yes or no? Yes or no? When you think about pigment, all of you think about color. Red color, green color, blue color, yellow color, brown color. But what is actually a pigment? Pigment actually is just a chemical compound whose electrons can be excited to the next orbital levels. Chemistry, physics, and biology. In order to understand photosynthesis, you need to know physics, chemistry, and biology. So what is a pigment? Pigment is a chemical compound whose electrons can be excited to the next orbital level. Amazing. Till here with me, all of you understood photosynthesis? We have just covered the first 10 lines of your NCERT now. Just covered 10 lines of NCERT. No doubt. Any doubt till now? Please ask me. Any doubt? Ask me right now. Shreyas sir was here. Any doubt? Ask me. No doubt? All of you are clear here? Now I'll tell you one more point here. Importance of photosynthesis is a general, very general thing. What is general thing? Primary source of food, I told you. Plant, we depend directly or indirectly on plants. Right? Directly or indirectly on plants. And responsible for release of oxygen. Right? Oxygen is released as a byproduct here. As a byproduct. Clear all of you? All of you know what is photosynthesis? Done? Now can we start with the experiments? Everyone in the chat should tell me. We just learned, sir, what is photosynthesis? In photosynthesis, reduction is happening. Anabolic process, endothermic reaction. Your carbon dioxide is getting reduced to glucose. All the points are clear. This session is going to be 3 hours. Maximum 3 hours. Because I have questions. I have questions today. PYQs. We'll be solving for PYQs. In detail, I'll be drawing each cycle. Amazing. Can we start with the next concept, that is the early experiments? The first part about photosynthesis is over. Requirements of photosynthesis is concept is clear in your brain, fixed in your brain, and we will go to the next concept now. Amazing to your notice. The next concept which I have to bring to your notice is, there was an experiment in your class 10th CBSE book. Right, class 10 CBSE book. In the chapter life process, you learned an experiment called as variegated leaf experiment in your class 10th. What was the experiment? The experiment told you that we have a leaf called as variegated leaf. This is a variegated leaf, means there are variations here. Variegated means variations in pigment concentration. There is a variation in pigment concentration here in variegated leaf. Can you see these are darker regions and we have lighter regions. In your class 10th itself you learned. In your class 10th you learned, if I keep this leaf, if I keep this particular leaf in a dark room, if I keep it in a dark room, every single starch is removed. But when I expose it to light again, only in the darker regions, starch accumulation has happened. What is the meaning here? And how do we do this? We did this with the help of iodine test. We know iodine will react with starch. In biomolecules chapter you have learned that iodine reacts with the starch, the amylopectin part. In this darker regions show that presence of starch. What do we understand by this experiment? This experiment shows us that chlorophyll is essential for photosynthesis. If you want to do photosynthesis, chlorophyll is important. Because only in this variegated leaf, wherever the chlorophyll was present, there and all you can notice the presence of starch. In this part there is no chlorophyll and hence no starch. Okay. Now this is only NCERT students. If you open your NCERT right now, you can find this experiment. That was just the basics which I want all of you to understand. Only and only NCERT. Okay. Keep your NCERT next to you. You can find this experiment right now. That is the second experiment that will show that light is essential for photosynthesis. This is only and only NCERT. Only NCERT students. If you, in this experiment, what happens? I'll tell you this. The second experiment in your NCERT. The second experiment which is given in your NCERT. Yes, whole chapter will be completed today. Second experiment in NCERT which says that you have an amazing leaf here. You have a leaf. NCERT is telling you in this experiment, I am covering, let me draw this green color here. Green. In this experiment, I am covering half of the leaf with a black paper, half of the leaf with a black paper. The rest of the part is completely free open. And then there is sunlight falling on it. Sunlight falling on it. And after some time, we did the iodine test. All of you know the iodine test. After some time, we did the iodine test, and what did we observe? When we do the iodine test, we observe that in the closed region, wherever it was covered, wherever the leaf was completely covered by the black paper, there is no starch. Why? Because the complete part of the leaf was covered. If there is no sunlight coming there, can we do photosynthesis? Can the plant do photosynthesis? Absolutely not. That is absence of starch in the parts leaf covered with black paper. Leaf covered by black paper. So what does the experiment shows? This experiment is showing you. This experiment is showing you that light is important. Light is important for photosynthesis. Are you with me, all of you, till now? Can I go to the next experiment? Is this experiment clear? Is this experiment clear? Can I go to the next very basic, students? If you read your NCERT right now, keep the book next to you. This experiment is very easy. Okay? So in the black region, wherever the black paper was covered, there is no photosynthesis. If there is no photosynthesis, there is no starch accumulation in the black region. Okay, let's go to the next experiment. The next experiment is your CO2 experiment. Okay, CO2 experiment. So we have a beautiful pot here, like all of you, the beautiful children. Similarly, you have a beautiful pot here. I love plants more than anything. Okay, so you have a beautiful pot here. In a second experiment, what did we do? We took a test tube. We took two test tubes. Let me draw here. In the second experiment, we take two test tubes. Two test tubes. In one test tube, I have a leaf. In second test tube also we have a leaf. Right? Second leaf. Leaf is there. Or keep an entire plant inside. That is better actually. Keep an entire plant. Small, kutti sa chotu sa cute plant you add inside. You add a small plant inside. And cover it with a cotton. Cover with cotton. Now here in this side, the cotton is there, right? Along with this cotton, you add something called as KOH. Can anyone in the chat tell me what is the meaning of KOH? It is none other than potassium hydroxide. Now this potassium hydroxide, which is present in the cotton, in this cotton, KOH is present. Now what this KOH will do, you know? This KOH is very cunning, very cunning. It will absorb, right? It will absorb all the CO2. It will absorb all the CO2 from the test tube. From this test tube, there is complete absence of CO2. Complete absence of CO2. But in this test tube number A, CO2 is present. And when you do a test later on, that is iodine test again, you notice that this particular leaf will have again starch, and this particular leaf will have no starch. No starch. Now what do you understand from this particular experiment? From this particular experiment you understand that CO2 is important. CO2 is very important to make glucose or starch. Clear? Simple? Done and dusted? Can we go to the next experiment? So I know students, some of you are thinking, sir, this is very basic. I know all of this. Why are you teaching such basic things? Because question will actually come from here, and then when the question comes right, I don't want you to be in trouble. Then you'll be like, oh my god, Basavaraj Sir said something I don't remember. You should not be like that. So I'm teaching you basics because not every student out there who knows all this right now. Because some of the students will be doing this chapter for the first time, right? For them all of this is, okay? Now, next one. Can we go to the next concept? First one was light. Yes sir, we learned light is important. Second, we learned about CO2 is important. The next one, the next scientist, This uncle right here called as Joseph Priestley. This uncle is so important because we know Newton discovered gravity. This uncle right here, very famous. What did he discover? Can you guess me? This uncle right here discovered your oxygen. He is the same guy who discovered the oxygen. Open your NCERT right now and you will find a line saying that Joseph Priestley discovered the oxygen. He discovered the oxygen. So he also gave you an experiment, okay? He gave you an experiment called as bell jar experiment. What does experiment say? This experiment is very simple. Look at the diagrams. You will understand yourself. Look at the diagram. So what do we have here? We have a candle. We have a mice. Okay. Don't do this. Don't, please students, disclaimer. Don't be like, sir, I did this experiment and then the mouse is dead. Don't kill innocent animals. Now we know. So don't do this. Okay. Don't do this. Okay. Don't do this experiment. Don't do it in your house. Okay. In this jar, the candle is burning. Whenever there is combustion, oxygen is used up, and this particular small rat or mice, it is doing respiration. Even in respiration, oxygen is used up. Yes, oxygen is used up. So whenever the complete oxygen is used up, the candle is gone. Rat is also gone. RIP everyone in the chat. RIP to your Mr. Rat. But Priestley did another experiment showing that he took a plant. He took a mint plant. Which plant, students? He took a mint plant, very tasty. All of you know I love, that is mint chutney. That is mint plant. He noticed that even after some time, the candle is still burning and the mouse is still alive. What is this magic? What is this magic, students? That is what Joseph Priestley said, that plants here are so special that actually plants are restoring the air. They're restoring the air. Whatever breathing animals, what did animals breathe? Animals were breathing oxygen. This candle was taking up oxygen. So what is your plant doing? Plant is replenishing the oxygen. It is taking up the CO2 and releasing the oxygen back. That is, and burning candle remove. So plants restore to the air whatever breathing animals and burning candle removed. What did they remove? Everyone in the chat who can tell me? The burning of candle and breathing removed CO2. Replacement was done by your plant which replaced it. Okay? That is replenishment of O2. That is your Joseph Priestley's experiment, bell jar experiment. Nothing fancy, nothing difficult. Done? Right? Done? Can we go to the next experiment? Any doubt here? In Tamil, Pudina is mint. Yes, in Hindi also. Kannadadalli, I don't remember. Okay? Can we go to the next concept? Clear till now, students? I'm teaching you very slowly because I want to have patience with all of you. I want all of you to understand each and every word, each and every experiment, each and every word I say today. Okay? Next one. The next experiment was given by your engine house, Jan Engine House. He did a similar experiment as your Priestley. Listen to my words. He did a similar experiment to your Priestley, but he did not do it on mid plant. He used a aquatic plant. And he kept this aquatic plant. Obviously, aquatic plants are inside the water. He did this experiment in the daytime. Whenever there is sunlight, the same experiment he conducted in the darkness. Sunlight and darkness. So what did he observe? He observed during the daytime around the aquatic plant. There was there was production of gas bubbles. And these gas bubbles are nothing but your oxygen bubbles. And same thing was completely absent in the night time. Completely absent in the night time. So here you can derive two things. What is first thing? First thing is the whatever reaction is happening, it happens only in the daytime and doesn't happen in the night. Means sunlight is important again. Sunlight is important. The second inference which we can make here is that your plants are producing a certain gas called as oxygen. Clear? Clear? All of you clear with this concept? All of you clear with this concept? Can we go to the next one? I know students, this can be little boring to all of you. Some of you thinking, sir, this is so easy. Why are you teaching over and over with so much of energy and enthusiasm? Because every single concept out here took so many scientists to do it. We are just learning it in one slide. But they did it such a long time. They did so many experiments. We have to actually pay respect to that. That is the reason we are doing this. So all of you pay respect to the scientists. Because without them, we would not be knowing any of this. Okay. So next concept. So here, what is the inference? Sunlight is essential for photosynthesis. Green plants of the part are release oxygen in the presence of sunlight. Okay. Done. Two of them are done. Amazing. Now, next one. Next scientist is called as, are all of you writing down? I believe all of you are writing down, right? Please write down this information because everything is important. Okay. Now, next scientist. Next scientist called as Julius von Sachs. Julius von Sachs. Now, he is a very interesting character because he was a scientist. For the first time, he told us that inside the chloroplast accumulation of sugar is happening. What else? He was a scientist who told us when plants grow, or do photosynthesis, or when they do photosynthesis, right, they produce something called as starch. They produce something called as starch. And he also told us that this starch is produced inside a structure called as chloroplast. It is produced in a structure called as chloroplast. And he was the same scientist who identified the chlorophyll. Who identified that chlorophyll. So he also told us that plants have a green pigment called as chlorophyll. He told us that plants have a green pigment called as chlorophyll. Clear? Clear all of you? Done? Remember, starch. What is, can I ask you a question here? Is this starch homopolymer or heteropolymer? First, who can tell me? This particular starch here, is it homopolymer or a heteropolymer? All of us know it is made up of your glucose monomers. It is a homopolymer. It has two parts such as amylose and amylopectin, which I will be teaching you in biomolecules, my second favorite chapter. Okay, so we'll wait for that. Is this concept clear? Is this clear? Amazing. Can we go to the next concept? Is this point clear? Next one. Now, from here things get interesting. You're almost getting end of your experiments. Okay, end of your experiments. Now here what do we have? We have something called as your Engelmann experiment. It's getting hot here. 1 second, students. Now let's understand Engelmann experiment. Can we start? So let's start this particular experiment with a question. Students, I hope this is not boring you because this is very important. I'm doing it very slowly actually. Which wavelength of light is optimum for photosynthesis? A simple question, right? Which wavelength light is important for photosynthesis? The answer was obtained when white light was sent through a prism. When white light was sent through a prism, we got your Vibgyor. Right? We obtained the Vibgyor here. He projected this Vibgyor on a algae. Can anyone chat who can tell me which is the algae he used? He used algae like your cladophora. Which is the filamentous algae? Cladophora. When this particular light was projected on the cladophora algae, he also introduced certain bacteria here. He introduced certain bacteria here. Yes sir, there are bacteria here. Which type of bacteria? He introduced aerobic bacteria. Aerobic bacteria. All of you know aerobic bacteria is a type of bacteria which will grow more wherever there is more oxygen. Yes, more oxygen. So he noticed that in this particular segment where your blue light, red is on top, where red light was falling, and this particular point where blue light was falling, in these two areas more number of aerobic bacteria had clustered. More aerobic bacteria had clustered in this particular two regions. What is the meaning of that? Meaning of that, more oxygen has been releasing there. Yes sir, more oxygen is being released here in these two areas. What is the meaning of that? Meaning of that is photosynthesis. Photosynthesis is happening at these two particular wavelengths, that is at 800 where red light is falling and blue color here at this particular two wavelengths where red and blue are light is there. Those particular location, photosynthesis is happening. That is the inference which we obtain from Engler experiment. And this also gave us the first action spectrum. The first action spectrum was actually provided here. The first action spectrum. Do not worry if you do not know what is difference between absorption and action spectrum. I will teach you in detail later on. But remember for now, the first action spectrum was obtained right here. Clear all of you? Any doubt here? Tell me right now if you have any doubt right now here. If you draw a graph for this, one. Any doubt here fast? Tell me. Wavelength and oxygen produced. We can see two different humps like this. This is for your blue. This is for your red. More oxygen is evolved towards blue side, less towards red side. So please remember, in the blue region, more photosynthesis is happening. More photosynthesis is happening in the blue region. Clear all of you? Till now, I hope I am teaching you most basic things. In entire chapter, students, the entire chapter also is like this. The entire chapter is also like this. There is no complication in this chapter. Such an easy, chota bacha chapter this is. Okay? Clear? Clear all of you? Can we go to the next react? Can we go to the next one? The next one is given by your, next experiment is given by your Cornelis van Niel. Cornelis van Niel was a microbiologist. He was a microbiologist who also did your botany experiments. So what did he tell us? Let's ask a question now. What contributes to the evolution of oxygen? I told you in the starting itself that whenever in a cell the hydrogen is split open, that oxygen is evolved. I told you this oxygen which comes out as a byproduct. The byproduct which is obtained out here actually comes from hydrogen, your water. But how do you know this? He did an experiment with a bacteria. You are amazing. So which reason less photosynthesis happen? That is your red, it is less, blue is more. Blue is more, red is little less. Okay? So how do we know this? He worked on a certain bacteria. He worked on some bacteria which used H2S. which use H2S as a hydrogen source. We know plants use water as for hydrogen source. Whenever I told you, when CO2 is getting converted to your C6H12O6, this hydrogen is obtained from your water. I told you. All of you know that, right? All of you know that. In plants, it comes from water. But in this particular bacteria here, they use H2S as the hydrogen source. What is this bacteria? This bacteria is nothing but your green sulfur bacteria, green sulfur as well as purple sulfur bacteria. These bacteria use your H2S, hydrogen sulfide, for the hydrogen source. So if I can write here something, let me write here. Let me use the pen, different color. If I can write a point here, that in your water, okay, this part is not, side is not visible. There's no space to move this side, but I'll write here. I'll write from left hand. In your water usually splits open into your H plus and O2, and this H2S splits open to H plus as well as SO4 minus. Now here can you see this sulfur? This sulfur is the byproduct. Released here, and in your plants, oxygen is the byproduct. So this particular scientist told us that H2O is a source of hydrogen in plants, and in the case of your bacteria, such as your H2S, in your bacteria as green sulfur as well as purple sulfur bacteria, H2S is the source of hydrogen. Clear till now? Any doubt, students? Any doubt till now? Any doubt? First ask me. All the experiments are over. All the experiments are over. All the experiments are over. Any doubt till now? First tell me. Any doubt till now? I will tell you radioactive genius. I'll tell you, and that's why I made this page. That's why I got this NCERT snip. That's why I got the NCERT snip of radioactivity. I will tell you that line here. I want all of you to focus here in this particular line of NCERT because this is a PYQ. So let's read. Can you read? All of you read with me? All of you read with me now. In green plants, H2O is the hydrogen donor. Yes, I told you, H2 in green plants, H2O is a hydrogen donor and is oxidized to O2. Yes, O2 is released as a byproduct. Some organisms do not release O2 during the photosynthesis, such as your green sulfur bacteria and purple sulfur bacteria. Okay, when H2S instead is the hydrogen donor for the purple and green sulfur bacteria. I told you the same. I told you this line. Yes, sir. The oxidation product is sulfur or sulfate depending upon the organism and not O2. So in the case of this bacteria, the byproduct which is releasing is actually sulfur or sulfate. Hence, he inferred that the O2 evolved by the green plants. The O2 which is coming from the green plants actually comes from where? The water. Comes from the water, not from the carbon dioxide. Now this was later proved by using the radioisotope technique, which someone was telling in the comment section. Yes, they use radioisotopes. Radioisotopes are like you have your 12, carbon 12, but some of them are 14. Now this type of carbon, right, 14 number, they can be tracked. We can trace them, like GPS location. We can trace them. How are they moving in a reaction? And that's how we later confirmed that the hydrogen comes from your water in the case of plants. Clear? I hope this part is clear to all of you. Should not have any doubt in all the experiments till we did that till here. You can crack NEET in 4 months? Yes, you can, and we will make sure it happens. Clear? Radioactive isotopes also clear? Now this is the final equation of your photosynthesis. That is 6 CO2 plus 12 H2O. In presence of light and chlorophyll, we get 6 glucose, that is getting converted to glucose, and your water and oxygen is the byproduct here. I hope all the experiments have been done. We have done each and every experiment in less than 1 hour, I guess. Students, the reason I told you so detailed, so slow, because I want all of you to understand this chapter. This chapter, how I told you this entire thing, right? I told you this entire thing. Entire chapter is so easy. So here they are telling you that oxidation product is sulfur, because H2S, when H2S breaks down, when H2S breaks apart, this hydrogen is donated for photosynthesis. Hydrogen is donated for your photosynthesis, for conversion of your formation of your glucose, and this particular sulfur is the byproduct. That is why your sulfur or sulfate, depending upon organisms. See, certain organisms are much more complex. See, this part is not in the NCERT right now. If you want me to teach, I can teach you. Okay? So if you want, I can teach you this, but this will go more. I have to show you 2 more reactions to teach you different organisms. That is, I have to teach you about mode of nutrition using chemicals. So that will take a lot of more time, which is not important for your NCERT or not important for your NEET examination. So let's stick to NCERT right now. If you want, I will make a small video on that in my Telegram channel. I'll upload it. Okay? Now let's stick to NCERT today. Now that was your experiments done and dusted. All the experiments done and dusted. Okay? Can we start with next concept? All of you ready? The site of photosynthesis. Can we start with the next concept, the site of photosynthesis? I know I'm going a little slow. This next concept will take 10-15 minutes. Then I'll teach you cycles. That is cyclic and non-cyclic. Half chapter is over. Then I'll teach you C3, C4, 3-4 chapter is over. Then I'll teach you Chemiosmotic theory. That is like 5 minutes Bacha chapter, Bacha concept. That is also over then. Then last concept is factor affecting. That is also over. Entire chapter is over. So students, I'll be teaching you the chapter in a very slow fashion so that you can understand each and every word I'm saying. Okay? Now, ready? I'm also ready. I forgot to get some water, but I'll drink some water later on. Now, let's understand site of photosynthesis. Can we start, students? Can we start? Okay, listen to me all of you. I told you before, photosynthesis happens in green plants. Students, do you want me to increase my pace? Do you want me to speed up more? Tell me in the chat. If you want me to speed up more, I can speed up. But if you want me to go in the same pace, where I teach you basic botany, basic terms, much more like as if you're learning for the first time, I'm teaching you. I am making sure, what I'm thinking is, all of you do not know anything about this. That's how I want to teach today. Okay. Photosynthesis happens in green plants, where in the leaf, on the leaf, we have special type of cells called as mesophyll cells. Yes, we have mesophyll cells. On this mesophyll cells, inside the cell, we have a structure called as chloroplast. Right? Chloroplast. That is the site. Chloroplast is the site for your photosynthesis. So I will draw right now. I will draw the structure here for chloroplast. So draw with me, understand with me, and we will start. Okay? Can we start a diagram? Now, here I will be teaching you small concepts, so please focus with me. Draw with me, all of you. This is your chloroplast. Can I tell this chloroplast is double membrane? That is, two membranes are here. Can I call this as outer membrane? Yes, sir, you can call this outer membrane. The inside part is called as inner membrane. Yes, sir, inside part is called as inner membrane. This is outer membrane. Okay. Now, two membranes are here. This is also like your lipid bilayer. Also like your lipid bilayer. Okay. Now, let's look at inside now. Now, inside your chloroplast, we have this coin-like structure. Draw with me, all of you. You have this coin-like structures. Can anyone in the chat tell me what are these coin-like structures? But because I will tell you in like a minute now, so let me draw. While I draw, I want all of you to start guessing. If you know, but if you do not know, don't worry, students. I am teaching this chapter for all the students who do not know anything about plant physiology. Zero knowledge about plant physiology to the advanced level of plant physiology. Okay. Now inside a chloroplast, you have this coin-like structures. Now each coin here. Now each coin is here is called as thylakoid. What is it called, students? Write down with me. It is called as your thylakoid. Thylakoid, right? Yes, sir. It is called as thylakoid. Now, what about this complete stack? What is this complete stack called as? This complete stack is called as grana or granum. Called as granum. Now, this entire stacks of grana is connected. It is the stack of coins, the entire stack of your thylakoids are connected by stomal lamella. Where do I mention this? Where do I have this? Stromal lamella. Clear till now? Yes, sir. Till now, everything is clear till now. Now I'll tell you one extra concept here. I want all of you to focus, but I will not teach you that now. I'll teach you later on that one. This is called as your thylakoid, granule thylakoid. Can I call this as granule thylakoid? On top of this granule thylakoid, I have two photosystems. Students, do not worry if you do not know what is photosystem. I will tell you what is photosystem later on. But remember for now, on top of this, but write down for now, on top of this granule thylakoid, we have two photosystems. One is called as PS2, other one is called as PS1. One is called as PS2, other one is called as PS1. On top of your stromal lamella, we only have PS1. We only have PS1. So students, don't worry, don't buy any of this. I will tell you in detail, I'll explain you what is the meaning of photosystem in detail. Wait for it. Okay, photosystem 1. Now, what about this matrix? This is matrix, right? This is the matrix here, like outer matrix. This is the entire thing is filled with matrix. Now, this matrix here is called as your stroma. It's called as what? Stroma. Now let me tell you one last point here. I want everyone to focus on this small last point here is on top of your thylakoid and your stromal lamella. On top of your thylakoid and stromal lamella, light reactions happen here. What happens here? On top of this and this, light reaction happens. Light reaction takes place here. And in the matrix, the stroma or matrix, dark reaction takes place. Dark reaction takes place. Clear till now? Till now, in this diagram, anyone has any doubt, I will move out of the way. You can take a screenshot, whatever you do, write down till here. If you have any doubt, please ask me right now. Please ask me. Any doubt till now in this diagram? I will tell you what is light reaction. Don't worry all these terms. Not for now. I will tell you while we go into the step by step. we are stepping into the water now. Our feet are just wet now. Now we are going to get deep into the concepts. Like similarly, you'll get into the concepts and I'll teach you today what is PS1, what is PS2, what is light reaction, what is dark reaction. Each one by one I'll teach you today. And after today's class, you'll be like, thank you so much, sir. This is the easiest chapter of all time. Okay? Okay, amazing. Now, I told you, leaf, mesophyll cells, chloroplast, outer membrane of chloroplast, inner membrane of chloroplast, stroma, which is the matrix. Each coin is called as what? Thylakoid. The entire stack is called as grana, and this connection between the two grana is called as stroma, lamella, and that's it. That's it. Okay? Till here, concept should be clear. This is also an NCERT diagram. Outer membrane, to show you I have not missed out anything. Outer membrane, inner membrane, stroma, lamella, entire called as grana. Each one is called as thylakoid. And then we have stroma, which is where the dark reaction happens. We have ribosomes. Then we have starch, granules, and lipid drops. And we have the lipid drops. Can we start the pigments now? Your next concept is over. Done and dusted. Next concept is pigment. Can we start? Can we start? Next concept is pigment. Can we start? Can we start with pigments now? I am teaching you most basic student. This is how everyone should learn botany. This is how I feel. Granular lamella. Listen to this. Where is it? The stromal lamella. Stromal lamella is the connection between the two grana. Clear? Clear? Starch means starch granules. Starch granules is nothing but storage of starch. Starch are like stored in a structure there. Okay? Now, what is the meaning of pigment? Can anyone in the chat tell me fast? Should I increase the pace? No, I don't think so, students. Learn this slowly. Learn it in detail today. You will have a lot of time to study this chapter later on, but stick with me today. You will learn this chapter. Okay, I promise you that. Now, what is pigment? I told you before. All of you think pigment is color, right? All of you think pigment means color. Blue color, red color, bottle green, blue color, green color, color, which color? Right? You think pigment is color? But actually, what is a pigment? Pigment actually is a chemical compound. I told you beforehand. It is a... Now, again, we are going to chemistry a little bit. Stick with me. I will teach you a little bit of chemistry now again. Okay? It is a chemical compound. It is a chemical compound. Yes, sir. It is a chemical compound which converts light energy. Into chemical energy. Right? Chemical energy. Light energy into chemical energy. Yes, sir. You told us before that. I also told you pigment. I told you in a pigment. Think this is a pigment. This is a pigment right here. Whenever a ray of light, that is photons, whenever photons land on your pigment, listen to me very carefully, students, now onwards. Whenever the photons land on this particular pigment, what happens? The electrons in that pigment get excited. The electrons get excited. The electrons are excited now. Electrons are excited. And these electrons are excited to the next orbital levels. And after some time, all of you know, the electron has to come down to the ground state. Yes, sir, it has to come down to ground state. So student, everyone should tell me in the chat right now. If you know chemistry, when an electron is coming down from a higher orbital level to the lower orbital level, what is happening? Fast, tell me in the chat, everyone. When an electron is coming down, and this has to be a fixed wavelength. Fixed wavelength. Obviously, not every wavelength will work. At a fixed wavelength, this is happening. Tell me fast, what is happening? When an electron is coming back when an electron is coming back to the ground state. When an electron is coming back to the ground state, see, this is all important, students. This is all is very important to understand this concept of photosystem later on. Okay, when the electron is coming back to the ground state, what is happening? It can emit light. All of you know in chemistry, we learned it can emit light. Can it also emit some that, what do you call it? I forgot the word. Heat. It can also produce heat. The third thing, when the electron is coming down, can it transfer the energy to another pigment? Yes, sir. When the electron is coming down, energy can be transferred. Electron is coming down, energy can be transferred to another pigment. Yes, sir. It can be transferred to another pigment. Now, the next concept here. What is the fourth step? One more thing which can happen. The one more possibility is that the energy which is coming down, you can utilize it for a particular reaction. Yes, sir. Some kind of reaction. The energy can be used for some reaction. This is actually called as photochemical reaction. Photochemical reaction. Yes, photochemical reaction. The first two concepts which we won't be studying here, that you will be studying in your chemistry, not my, I won't be teaching you chemistry right now. But these two concepts here, these two concepts right here, we will be studying today's video in photosynthesis. Ready? Ready with me, all of you? These two concepts, that is energy transfer to another pigment, that we'll be studying today in today's video. In today's chapter, we'll also be learning about photochemical reactions. Whenever the electron is coming down, that energy can be utilized for certain photochemical reactions. Certain photochemical reactions. Clear? Now, before I go to type of pigments, now can I teach you some physics? Before I go to the next concept, can I teach you some physics? Ready for some physics? Students, if you want to understand the concept of photosystem, right? If you want to actually understand the photosystem concept, you need to know a little bit of physics also. Don't by heart all this. Don't by heart all these concepts. Don't by heart that, electrons are just jumping. Why are they jumping? Understand the concept. Okay. So, bear with me for a little physics now. Can I teach some physics? Yes, sir, you can teach some physics. So, we have your pigment here. On this pigment, light is coming. Say this pigment it is getting excited at 600 nanometer. It is getting excited at 600 nanometer. Okay, 600 nanometer. Now, this is getting excited. What is released? Electron is ejected. Electron is ejected to the next orbital. Right? When this is coming down, when electron is coming back to the normal state, energy is being released. And energy is being released. Do all of you know this? Energy is inversely proportional to lambda. Do all of you know this? Energy is inversely proportional to lambda. Yes, sir. Energy inversely proportional to lambda. So imagine we have multiple pigments here. Different pigments. This pigment is getting excited at around, take it as around 640 nanometer. This is getting excited at around 550 nanometer. This is getting excited at around 520 nanometer. Now I told you in the previous slide, whenever the energy is coming down, we can transfer the energy from one pigment to the next pigment. Third possibility. Yes, sir, you're with me right now. All of you are with me? Yes, sir, all of you are with me. Right, all of you with me. Energy can be transferred to another pigment. That's what we are doing here. That's what we are doing here. We are transferring the energy to another pigment. Now, who can tell me where does the energy go? The energy will be transferred to this wavelength. This wavelength. Why? Why to this wavelength? Because energy is decreasing. As electron is coming down, energy is decreasing. So always remember whenever the electron is coming down, energy is decreasing. So the energy, it will always will go to the highest wavelength. It will always go to the highest wavelength. Now this concept is actually important. Where do you utilize this? Where do you use this concept? In your non-cyclic phosphorylation. In non-cyclic, all of you, some of you who know that from PS2, I'll be teaching you, don't worry. Some of you know that from PS2, the electrons are transferred to PS1. All of you know that. We also know that PS2 is at 680 nanometer and this is at 700 nanometer. That is why, remember, energy can always be transferred from lower wavelength to higher wavelength. That is why in your non-cyclic phosphorylation, which I'll be teaching you later on in detail, line by line, that is why energy is getting transferred here from 680 to 700. 680 to 700. Clear? All of you clear? Can we go to the next concept? Physics is done. Physics is done. Chemistry is done. Sir, come back to biology now. Can we start with bio again? Can we start with bio? Let's start with bio now. Clear? Now let's start with biology. That is the next concept. Biology, the next concept which I have to teach you is, I'll not teach you what is paper chromatography. Next I'll be teaching you types of pigments. I'll be teaching you types of pigments now. Students, I want hearts, fire in the chat, because this detailed session, right? This detailed, slow pace, every single point I'm taking up from NCERT and teaching you the photosynthesis in detail today. Okay? Can we start? Can we start? Now, NCERT is mentioning a particular type of technique called as paper chromatography. What do we do in paper chromatography? In paper chromatography, we take leaves, we crush the leaves with the help of mortar and pestle. With the help of mortar and pestle, we completely decimate the leaf. Then we add a solvent. After adding a solvent, we use a Waterman paper, filter paper to filter out the liquid. Yes, liquid is filtered out. Now this particular liquid has your different types of pigments and we load it onto here. We load here and then we keep it in a solvent. And based on the, I won't teach you the principle right now, based on your retention, that is, principle is later on, principle is not important for you, concept is important. Based on the retention, your different pigments are separated, such as your chlorophyll B, chlorophyll A, xanthophyll, as well as carotenes. Xanthophyll and carotenes, these are the different pigments which is given in your syllabus, and to extract them, to separate them, we use a technique called as paper chromatography. Paper chromatography. So what do we have? We have chlorophyll A, chlorophyll B, xanthophyll and carotenes. Okay? No, this won't be 6 hours. I'll finish it in 4 hours maximum, but I want to finish in 4 hours. Not more than that, because I don't want to drag it. Okay? Clear? Now, till here is clear. Because next I'll be teaching you what is absorption spectrum as well as the action spectrum. Ready? Is this point clear, all of you? Is this point clear to all of you? Can I go to the next concept? Can I go to the next concept, all of you? Paper chromatography is a technique to separate the pigments such as your chlorophyll A, B, xanthophyll and carotenes. Okay. Now, let's understand what is the meaning of your absorption spectrum. What is absorption spectrum? Now, this pigment you take, right? This pigment you take, you run it on the machine. When you run it on the machine, we get a spectrum like this. So what is your absorption spectrum? Absorption spectrum is a spectrum like this, which shows us, what is it showing us? At which wavelength, which pigment is getting absorbed. Listen to my words. Absorption spectrum is a spectrum which is showing you at which wavelength which pigment is getting absorbed by the light. Clear? So I'll repeat, I'll show you once again this one. If you don't understand, I'll tell you once again. We have a white light coming here. Imagine I'm putting a white light. I'll explain again one more time. If you don't understand, this is your pigment here. Pigment is here. I am adding a white light. White light is here. White light is here. Now something is coming out here. We have light which is not absorbed light is coming out. Not absorbed light is coming out. The not absorbed light is coming out. But can I tell some form of light has been absorbed by the pigment? Some amount of light has been absorbed by the pigment, and whichever light is absorbed at what wavelength, that is your absorption spectrum. That is your absorption spectrum. See here, pigments have ability to absorb light. I told you pigments can absorb light. The absorption spectrum of leaf pigments indicates that the wavelength, at which wavelength light absorbed by the each pigment. So each pigment here, carotenoids, chlorophyll B, chlorophyll A, all the pigments are absorbing some light. At which wavelength, that is called as your absorption spectrum. That is called as your absorption spectrum. Clear? 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 Just say yes sir. Absorption spectrum is clear to you. Absorption spectrum is clear to all of you. Clear? Now one more thing which is a NEET PYQ is this point here. Maximum absorption by chlorophyll A is in the blue and red regions. I told you in experiment before, the prism experiment, English experiment. I told you the experiment, remember that blue and red are absorbed. Blue is more, red is little less. Red is little less, but blue is more. That is nothing but your absorption spectrum, at what wavelength a pigment is getting absorbed. Clear? Now what is action spectrum? If you want to learn action spectrum, just look at the action which is happening here. What is the action which is happening here? What is the meaning of action? Action means work is done here. Yes, work is done here. What is the work here? The work here, which we represent here, is photosynthesis. Photosynthesis. Work is photosynthesis. Change this. Photosynthesis or O2 evolved, or photosynthesis or O2 evolved. At what wavelength? At which wavelength photosynthesis is happening? That is your action spectrum. At which wavelength here? At which wavelength photosynthesis, the work is done, the action is happening. The action is happening at which wavelength is called as action spectrum. So action spectrum shows us, what does it show us? It shows us the action. It shows us the photosynthesis happening at which wavelength. Can you see one hump here? That is for blue. This is the action spectrum of your chlorophyll A. This is blue region. This is the red region. This is the blue region. This is the red region. At these regions, action is happening. Photosynthesis is happening. Clear? Do you want me to repeat this again? Students, do you want me to repeat this again? Tell me first. Do you want me to repeat this experiment again? See here. The graph indicates the wavelength of light. Right? The graph is showing us, the action spectrum is showing you at which wavelength photosynthesis is happening. That's all. That is your absorption spectrum. Absorption spectrum is nothing but at which wavelength photosynthesis is happening. See, I cannot tell more easy than this. This is the easiest definition of action spectrum. The higher the rate of photosynthesis is in the blue and red region for your chlorophyll A. This is the blue region. This is the red region. Can you see red? This is the blue region. At the blue region, red region, photosynthesis is happening, action is happening. Thank God you understood. See, I have faced this difficulty. When I was teaching in offline conditions, this particular concept was so students used to suffer. I still know so many students who do not know difference. Okay. Now, let's superimpose them. Let's keep that one above the other. When you superimpose your action spectrum as well as absorption spectrum, do you see the similarities? Yes sir, I see similarity. Whenever there is absorption, whenever there is high absorption of particular wavelength, at that wavelength only photosynthesis is happening. Yes sir. If you look at this peak here, at this peak absorption is happening. Yes sir, at this peak red light is getting absorbed. At the same peak itself photosynthesis is also happening. That is the comparison. That is the comparison. But if you notice one difference here, this particular point here, at this particular point, even if you open your NCERT right now, you will find at this point there is no absorption. At this point there is less absorption, very less absorption. But what is the but? But photosynthesis is happening. Photosynthesis is happening at this particular point here. Absorption is not happening. For chlorophyll A, absorption is not happening. But photosynthesis is happening. How? Who can tell me in the chat? If you open your NCERT right now, I wish I had that image. This image is not good. If you open your NCERT right now, they have given you an image which shows superimposition of your action spectrum and as well as your absorption spectrum at one particular level. When this is your graph, this is your hump here. This is your absorption. There is no absorption happening here. But if you look at this, your action spectrum will look like this. Action spectrum means what? Photosynthesis is actually happening here. What is the meaning? The answer is very simple. Even though chlorophyll A is not getting absorbed, the pigment is not getting absorbed as wavelength, but other pigments, such as your chlorophyll B, carotenoids, xanthophyll is getting absorbed, and all the energy absorbed by these pigments called as accessory pigments, the energy is sent to your chlorophyll A. All the energy is sent to the chlorophyll A. Let me draw it. This is a very important concept which so many students miss out. At this particular point, chlorophyll A is not getting absorbed. Listen after me, students. At this particular point, chlorophyll A is not getting absorbed. Chlorophyll is chill, bindas. But accessory pigments, the goons, the gundas, are separate. I'll tell you a story later on. The accessory pigments are very active because that's their wavelength. They are absorbing the energy. But can they do photosynthesis? Can your accessory pigments do photosynthesis? No, because they do not have the enzyme. In fact, all the accessory pigments are sending the energy. All the accessory pigments around it. All the accessory pigments are sending energy to your chlorophyll A. And this chlorophyll A right here is doing the work. That is why in this particular dip, even though there is no absorption, but work has been done. Clear? Yes, it does not completely overlap. It will not completely overlap. There will be minor changes, obviously. There will be minor changes here and there. Is this concept clear? All of you? Is this concept clear to all of you? Why? Even though there is a dip, accessory pigments are in work. Because your accessory pigments are working still. Accessory pigments are working. Because at this wavelength, chlorophyll A is not active. Chlorophyll A is not getting absorbed. Chlorophyll A is not getting absorbed. But other accessory pigments are absorbed. Their absorption spectrum is that. Okay? Amazing. All of you are amazing. All of you understanding. Now, this is the question. Rate of photosynthesis depends on action spectrum. Action spectrum is actually showing you where the photosynthesis is happening, not the rate. Action spectrum is showing you at which wavelength photosynthesis is happening. Okay. Now, I told you there are photosynthesis pigments. We have something called as your chief pigment. Chief pigment is the boss. In your movies, you have noticed, in the movies, there is a main boss and there are small gundas. Main boss and small gundas are there. Yes. Main boss and small gundas. The main boss is called as your chlorophyll A. Is a chief. Chlorophyll A is the main boss. And the small minions, the small gundas are nothing but your accessory pigments. Clear? Now, last year in the NEET PYQ, last year students, there was a NEET PYQ based on this information. There was a NEET PYQ based on this information here. What is the information here? Chlorophyll A is blue-green. Chlorophyll B is yellow-green. Carotenoids is yellow to yellow-orange. And xanthophyll is yellow. Do you want a trick to remember this? Do you want a trick to remember this, all this information? Do you want a trick? I'll tell you a trick. See, I usually rarely tell a tricks because I believe in pure knowledge. Tricks are always secondary. But all of you should understand the concept. Right? All of you should understand the concept. But tricks, today I'll tell you a trick. Can anyone tell me what is Vibgyor? This is your Vibgyor. Right? Vibgyor. The first one is your chlorophyll A. The first one is chlorophyll A. Right? First one is chlorophyll A. Chlorophyll A is between your blue and green. Yes, sir. Chlorophyll A is between blue and green. Now, what about chlorophyll B? Chlorophyll B is second between yellow and green. So, can I say yellow? This is yellow and green. This is chlorophyll B. This is chlorophyll A. Next one is your carotenoids. Carotenoids is between yellow to orange. So, this is yellow, right? Why? Yellow to orange is your carotenoids. And the last one is xanthophyll, that is your yellow to orange. Yellow to orange it should be. Yellow to orange. Yellow to orange is nothing but your xanthophyll. Clear? Clear? Is the trick clear to all of you? Vibgyor, blue-green, chlorophyll A. Yes, sir, blue-green, chlorophyll A. Green and blue. Sorry, yellow and green. Right? Yellow and green. Yellow and green is chlorophyll B. Next one. Then we have carotenoids, that is yellow to yellow-orange. Yellow to orange is carotenoids. Then lastly we have the yellow to orange, that is yellow to orange is xanthophyll. Clear? Amazing. So students, if you know this Vibgyor, if you knew this Vibgyor, this was a NEET PYQ last year. Last year, the most basic question they're asking you from this NEET 2023 question, this is. It can be asked again. Okay. So, done with the concept. Next concept. Now, the next concept which I want to teach you is classification of pigments. I told you we have chlorophyll A. We have chlorophyll A, then we have main pigment is chlorophyll A, then we have auxiliary pigments. I told you we have many auxiliary pigments. But students, listen to me right now. I will teach you what is the meaning of LHC, light harvesting complex. Okay, now let's start the next concept. Imagine we have your I have to teach you what is reaction center. Okay. I told you there's a main boss. I told you there is a main boss. I call this main boss chlorophyll A. I call this main boss chlorophyll A. Now this chlorophyll A absorbs a wavelength at 680 nanometer. Main boss absorbs wavelength at 680 nanometer. Okay, listen to me very carefully now, students. Next 5 minutes give me. I will teach you what is the meaning of photosystem, what is the meaning of your LHC, light harvesting complexes. Okay, main boss is your chlorophyll A. Then we have small bosses, small gundas. When we have small gundas, such as your chlorophyll, can I call this chlorophyll B is your small gunda. Then we have even more small gundas like this, called as your carotenoids, called as your, let's take a xanthophyll. Then we have even more smaller gundas, called as your carotenoids. Now let's say the carotenoids absorb the wavelength from your 500 to 550 nanometer. Carotenoids absorb the wavelength from 500 to 550 nanometer. Then we have xanthophylls. Xanthophylls absorb from your 550 to 600 nanometer. Then we have your chlorophyll B. Chlorophyll B will absorb from your 600 to 660 nanometer. Yes, till now it is clear? Till now everyone with me? Till now all of you with me? Yes sir, all of you with me? Whenever there is a light coming in, whenever there is light is coming in, yes sir, light is coming now. Now this light will have something called as PAR. All of you know what is PAR? Photosynthetically active radiation. In your ecosystem you learn it. In your ecosystem you chapter you learn it. PAR, which ranges from your 400 to 700 nanometer. Yes sir, all the concepts are corrected. This is in ecosystem. PAR, photosynthetically active radiation. Now sunlight is coming. Now can I tell some amount of sunlight is taken up by this small gunda here. Small gunda is taking some sunlight. Now this small gunda is small gunda. Why? Because it does not have the power to do photosynthesis. The small, all of these are called as the small gundas and all the other thing your accessory pigments, right? Accessory pigments. Now these accessory pigments, right? These accessory pigments cannot do photosynthesis. All they can do is transfer the energy. So whenever a light is coming, the energy is getting transferred. The energy, they just transfer to the next level. They just transfer the energy. They just transfer the energy all the way back to the chlorophyll A. They transfer the energy to your chlorophyll A because chlorophyll A is the main boss who can take action, who can do photosynthesis. Are you with me till now? Are all of you with me till now? Any doubt, any confusion, ask me right now. I know the story is getting complex, right? The small gundas are called as your accessory pigments. They cannot do photosynthesis. All they can do is capture the light. They are sidekicks. They can capture the light. They can only transfer the light. Yes sir, they're transferring the light to your main boss. They're transferring the light to the main boss, and the main boss will be the highest. Always remember, the main boss is the highest wavelength. Main boss at highest wavelength. Okay. So why am I telling you all of this information? Because you need to know the function of accessory pigments. Accessory pigments has two main functions. What are the two main functions? Can anyone in the chat tell me? So explain the last line. So last line which I'm trying to tell you here is your all the gundas, right, small accessory pigments, they only capture the light and they transport the light. Sorry, transport energy. They capture the light at this wavelength. At these wavelength, They capture the light and they transfer the energy. Why are they transferring the energy? Because they cannot do photosynthesis. They cannot do photosynthesis. So what is the function of axillary pigments? I've told you. Not told you, I haven't told you, right? I'll tell you now. The function of your axillary pigments is, write down students, the first function which is given in your NCERT without explaining. The first function is widening. Widening the range of wavelength. Just like in your movies, can the main boss go to all small local areas and can he show his power? No. He has small gundas everywhere to go to each area and do the work. Similarly, here also, the main boss has small gundas. What will they do? They will widen the range of wavelength at which light can be absorbed. That's why we say PAR is from 400 to 700 nanometer. We have the main boss has control over entire region. How? Because of the small gundas. They widen the range of wavelength. Wavelength has been absorbed, increased, so more light can be absorbed from everywhere. And the second one, the second important point here is whenever the main hero goes and attack the villain, don't you remember in the movies, the small villains will come first and protect the main villain. They'll protect the boss. That's what they're doing. The second function is protection. That is protection against oxidative. The protection against chlorophyll, for prevention, protection against photooxidation. Protection against photooxidation. Just like in the movies, the main boss is protected. Similarly, the main boss here, the chlorophyll A, is protected from all the photooxidation. Clear? Clear? I hope this concept is clear. The function of your axillary pigments. Can I erase this? Can I erase the story? Can I erase the story? All of you know the function? All of you know the function of your axillary pigments. What is the function? Widening the range of wavelength at which we can absorb the energy. Yes, we have widened the range of absorption. And the second point here is protection against photo oxidation. Clear? So I hope this concept is clear to you, because NCERT is telling you this. NCERT is mentioning this word. If you open your NCERT, it is telling you the function of your axillary pigments. Okay. Now can I classify? Can I erase the students? Can I erase this? Yes. Can I classify the pigments based on the function into two types? Can I classify the pigments based on two types? The first one, the main gunda, called as your chlorophyll A. Can I call it, also call it reaction center? Can I call it reaction center? At this particular reaction center, the main work is done. That is, photosynthesis is happening. Photosynthesis is happening here. This is the reaction center, because here we have a lot of enzymes. We have a lot of enzymes here. That is chlorophyll A, reaction center, main boss. Lot of enzymes are here that can do. The second, the small chotu bachas, the small gundas. Can I call them antenna? Can I call them antenna? Can I call them antenna? Why are they calling the antenna? Because all they do is transfer energy. Transfer energy to the chlorophyll A. They transfer the energy to the chlorophyll A. So antenna does what? Antenna captures all the energy. I showed in the picture. They will capture everywhere. They'll widen the range. They capture the energy and they transfer to your chlorophyll A. Clear, sir? That is your, what do we have here? Here we have your chlorophyll B. We have carotenoids as well as xanthophyll. Yes, we have xanthophyll. All of these are here. Yes, sir. This is also correct, sir. All you're telling is correct. But why are you telling all this? Students, this is nothing but This is nothing but your LHC. What is LHC? LHC is nothing but your light harvesting complex. Light harvesting complex. Light harvesting complex. What is the meaning of light harvesting complex? They are harvesting light. First, main boss is, so see, remember students, main boss can also harvest light. Main boss can also do harvest light. He can also fight. Why? Because it has pigment. It has wavelength. This boss will work at 680 only. Only 680 nanometer will work. But this is antenna which will work on a wide range. So this is nothing but light harvesting complex for you. This is light harvesting complex. So don't by heart, understand the concept. Okay? Ready all of you? Can I teach you what is photosystem now? Can I teach you what is photosystem, all of you? Tell me in the chat. All of you understood what is? After I teach you photosystem, I'll take a 5 minutes break. After teaching photosystem, I'll take a 5 minutes break. Then we'll start your cyclic and non-cyclic. Okay? All of you understood what is the meaning of LHC? Light harvesting complex. Clear? Okay. The next concept I'll teach you is there was space here. I didn't have to erase it. I had a space here. The next concept I'll teach you is photosystem. Okay? All of you are enjoying? Are you enjoying botany today? Are you learning apart from textbook what is given in textbook? This is how it actually is, sir. Okay, this is how actually botany is. So concept of photosystem. Photosystem is nothing but photosystem is nothing but your LHC, light harvesting complex. What is present in light harvesting complex? Your reaction center as well as antennas, certain electron carriers, and also certain enzymes. Now what is electron carriers? For example, in your PS2, we have your pheophytin, right? We have pheophytin there, and in the photosystem 1, we also have your sulfur centers. These are your electron carriers. So what is photosystem? Photosystem is made up of your LHC, electron carriers, and enzymes. Where are they present? They are present on your thylakoid membrane and also on your stromal lamella. Clear? All of you know what is reaction center now? All of you know what is reaction center? So in your syllabus, not your syllabus, in general, we have two reaction centers. In general, we have two photosystems. In general, we have two photosystems, such as your photosystem 1 and photosystem 2. Okay, photosystem 1 and photosystem 2. Now, what is the difference here? Yes? Clear till now? All of you following me, right? All of you following, or did I lose all of you? Did I lose all of you, or are you still with me? Tell me, students. I have built up the chapter now. We have building up the foundation. From foundation we went from your small pigment, we went from your photosynthesis, pigments, photosystem. So we went to your antenna, big boss, small boss. Finally we are here now. Finally we are at the photosystem. So we have two types of photosystem. We have one is called as photosystem 2. We have photosystem 2. The photosystem 2 works on, absorbs the wavelength of your light of your 680 nanometer, while your photosystem 1 absorbs the wavelength of light at 700 nanometer. Clear? Clear till now? Is this concept clear? So can I tell what is the other difference here? The other difference here I have to tell you is photosystem 1. Why is it called as 1? Because it was discovered first. Photosystem 2, it was discovered later on. It was discovered later on. That is second. Clear? They are present on your thylakoid membrane. On thylakoid membrane, we have photosystem 2 and photosystem 1. I'll tell you the difference again. It can absorb the wavelength of light at 700 nanometer, that is 1. What about 2? It can absorb the light at 680 nanometer. Yes, it is a last year PYQ. This right here is a last year PYQ, all of you. This was discovered first. This was discovered second. Reaction center. It is specialized in chlorophyll A, which absorbs the light of 700 nanometer. It has specialized chlorophyll A, which absorbs the wavelength of light at 680 nanometer only. 680 nanometer only. And if you remember my chloroplast diagram, we had your thylakoid membrane and thylakoid membranes. On your thylakoid, on your granule thylakoid, we have both photosystem 1, photosystem 2, as well as photosystem 1. And on your stroma lamella, we only have what? We only have your PS1. We only have what? PS1. We only have what? PS1, that is at 700 nanometer. Clear? Yes, it is based on discovery. I told you, right? I told you based on discovery. This was discovered first. This was discovered second. That's why it's called as photosystem 1. That's why it is called as photosystem 2. Now, phase of photosynthesis. In photosynthesis, the entire photosynthesis is divided into two parts. That is light reaction. Finally, we are coming to the main concept here. Students, we are coming to the main deep concept now. The core concept. After 2 hours of photosynthesis in higher plants, we are coming to the main deep concept. That is your light dependent reaction, called as light reaction. The other one is called as dark reaction. Now students, a simple question. Does the dark reaction happen inside darkness? Does the dark reaction happen in darkness? Can anyone tell me? Does dark reaction happen in darkness? Absolutely not. Dark reaction, why is it called dark reaction? Because it is dependent. It is dependent on the products of your light reaction. It is dependent on the products of light reaction. That is why it's just called as dark reaction. It does not happen in the darkness. Dark reaction also happens in the daytime. It also happens in the daytime. Clear? Can we start now? Light reaction. That is occurs within one more thing. Where does it light reaction happen? Light reaction happens on your thylakoid membrane. That is occurs within the membrane of the chloroplast. Thylakoid membrane happens in stroma lamella. Trapping of light energy by pigments. I told you pigments. Now we know pigments, they trap the energy. Then it synthesizes ATP and NADH. I will teach you all this. Don't need to remember all this. I'll teach you one more time all this. How many ATPs, how many NADH, I'll tell you. And in the case of your dark reaction, it is synthesized in the where? In the stroma. It is dark reaction is happening inside the stroma, and ATP and NADH are used here. They're what? ATP and NADH are not produced here. They are used here. So whatever ATP and NADH produce here, right? The same ATP and NADH are used in the dark reaction. They are used in the dark reaction. Clear? And in light reaction we get ATP and NADH. But what do we get in dark reaction? In dark reaction we get your sugar. You get your sugar. That is the main reason, right? You're doing photosynthesis. You get obtain sugar in your dark reaction. In your dark reaction. Clear till now? The next one concept I'll tell you is in light reaction 2 pathway. One is non-cyclic photophosphorylation. What is the meaning of phosphorylation? Phosphorylation is addition of phosphate. In photo means in presence of light, but non-cyclic. The other one I have to focus on is cyclic phosphorylation. The other one is called as cyclic photophosphorylation. Till now with me? Till now are you with me? In light reaction, we have two types, non-cyclic and cyclic. Can we take a 5 minutes break now and start? Can all of us take a water break, rehydrate yourself, and please come back. Students, till now was the foundation. Till last 2 hours, almost 2 hours, was just the foundation. Now, from this point onwards, the most amazing topic, the most easy bacha topic. See, if you understood the concepts till now, this is very easy. This is very, very easy. Okay, so we'll take a 5 minutes break. It is your 8:20, right? 8:20, 8:20, 8:21. Let's start at 8:30. 10 minutes break we can take. Can I start at 8:30? Can we start at 8:30, all of you? Break till 8:30. Can we take? Amazing, students. But I want all of you to come back. Do not leave at this point. See, you have invested 2 hours with me right now. Your life's 2 hours you have given to me. Please give me 2 more hours of your life, and I'll make sure this chapter is imprinted in your brain. Okay? Okay? Amazing. You have invested so much of time. I want it to be useful now. Okay? So can we start after 2 hours? Okay. So after 10 minutes, not 2 hours, at exactly at 8:30, we are starting. Okay? So see you at 8:30. Bye-bye. Sayonara. Can we start? Is the time already? No, we have a minute. So can we start, students? I just noticed this light is coming. Is this light here? I don't even know where this light is coming from. The reflection is coming. Is this? No idea. Absolutely no idea. Who cares? So break over. So till now, what have we learned? Let's summarize what have we learned till now. We started off with basic definition of your photosynthesis. Yes, sir, we learned about what is photosynthesis, what is happening in photosynthesis. Then we climb a little higher. Then we learned about what are the requirements of photosynthesis. Reduction process, it's an endothermic process and it is an anabolic process. Then we learned on to different early experiments which shows us the light is important, carbon dioxide is important, oxygen is present, right? All different experiments we saw. Whenever the oxygen requirement, the hydrogen comes from your water. Then we learned about pigments, different types of pigments, like your main pigment, that is your chlorophyll A. Then we learned about your accessory pigments, such as your carotenoids, chlorophyll B. Then we also learned about your xanthophyll. Then we also learned about your what we learned? Action spectrum and absorption spectrum. Then we learned about LHC, light harvesting complexes. Then we learned about photosystem. We also learned about your structure of your chloroplast. So till now, I hope none of you have any doubt till now. Amazing. Can we start? Can we start the next part of the class? Can we start with the non-cyclic phosphorylation? Non-cyclic photophosphorylation. Can we start, everyone? Can anyone in the chat tell me thumbs up, hearts, fire? I need to know that all of you are ready. What are xanthophylls? Xanthophylls are a type of your pigment which produces your yellow to orange color, right? Amazing. Till now, I believe none of you have any doubt, and I will start in the next topic, that is non-cyclic photophosphorylation. Big terms, heavy terms, but it is very, very easy. Can we start? Amazing. I have all of your permission, and I will start with the simple diagram. I want all of you out there, please write down the diagram with me. Okay, write down the diagram with me. It will be useful to you only, not for me. Okay, so write down the diagram with me. First, we have the main boss here. So I told you, in non-cyclic phosphorylation, where does it happen? Where does it non-cyclic phosphorylation, when does it happen? The question is very simple. The answer is also even more simpler. Non-cyclic phosphorylation happens when a wavelength of light less than 680 nanometer is absorbed. Agree with me? All of you should agree with me. Non-cyclic phosphorylation will happen only and only when a light wavelength of less than 680 nanometer is absorbed. Why? Because your photosystem 2, here your photosystem 2 absorbs the light of wavelength 680 nanometer. If the wavelength is more than 680, can it absorb the light? Can it absorb the light? No, it cannot absorb the light. Yes, it cannot absorb the light if it is more than 680 nanometer. Now what is this? This is your chlorophyll A reaction center. Chlorophyll A reaction center. Now all of you know, along with chlorophyll, we have many other pigments called as accessory pigments. Yes, sir. So can I draw multiple accessory pigments here? Accessory pigments. Yes, sir. We can draw. Wherever a wavelength of light is coming, photons are coming from the sun. photons are coming from the sun. All these photons, the light, PAR, has wide range. All the wide range of wavelength is utilized and the light is absorbed. Yes, all the small minions here absorb the light and send the energy. They send the energy to your chlorophyll A. Yes sir, they are sending the energy to chlorophyll A. Chlorophyll A is also very active. It is also absorbing the light. Yes sir, it is absorbing light. Whenever it is absorbing the light, I told you, pigment is a chemical compound which can get excited. Yes, it can get excited. Right? It can get excited. So this one is getting excited here. It's got excited. Now this is getting excited and the electrons are shot up. 2 electrons are sent up. Please write down with me. 2 electrons are shot up. Now these electrons are accepted by a primary electron acceptor. By a primary electron acceptor, which is none other than in non-cyclic phosphorylation for your photosystem 2. For your photosystem 2, it is none other than your pheophytin. It is pheophytin. It is pheophytin. Pheophytin absorbs the electrons now. Now from here onwards, a series of electron transport chain is happening. Electron transport is happening. Yes sir, a series of electrons are getting transported here. Now from here, from your primary electrons, from here it goes to PQ. What is PQ? PQ is nothing but your plastoquinone. Right? Plastoquinone. Plastoquinone. From plastoquinone, the electrons are transferred. Electron transferred to your cytochrome. To your cytochrome B6F complex. Write down students all this. It's sent to your, I'll write down the full forms here somewhere later on. What is PQ? PQ is plastoquinone. What is CY? CY is nothing but your cytochrome B6F complex. Now from here it goes to your PC, not your personal computer. It goes to your plastocyanin. It goes to your plastocyanin. Yes sir. So can you see a downhill movement of electrons? Yes sir. The electrons downhill is happening because energy. The energy is being reduced. Energy is lost. Energy is lost. Now from here, from PC, the electrons are sent to your next reaction center. The next reaction center, that is your chlorophyll A again. The PS1 reaction center that works at 700 nanometer. At 700 nanometer. All the electrons from here, they sent to your chlorophyll A. Now from here, again the electrons are getting ejected because it's also pigment. I told you in the starting. Remember, electrons can be transferred from one pigment to next pigment. I told you, right? Now from your lower wavelength to higher wavelength, I taught you that concept also. Why? Because energy is inversely proportional to lambda. Yes. So lower wavelength to higher wavelength because energy is getting decreased. Now from here, electrons are again shot up. Again, electrons are shot up. Now again, from here, electrons are accepted by your primary electron acceptor. Primary electron acceptor. Now who is the electron acceptor here? The electron acceptor here is none other than an iron sulfur centers. Iron sulfur centers. FES. Iron sulfur centers is your primary electron acceptor for your chlorophyll A. This can be asked as a separate question. Asked as a separate question. The primary electron acceptor for chlorophyll, that is photosystem 1 is FES. For photosystem 2 is your pheophytin. It is your pheophytin. Now from here, from FES, it goes to your FD, ferredoxin. From ferredoxin. Now from ferredoxin, the electrons, the final electron acceptor, The final electron acceptor is none other than in your outer membrane. Can anyone in the chat tell me who is the final electron acceptor here first? The final electron acceptor is your NAD plus. NAD plus, guess it is taking up the electrons, gaining the electrons, is getting reduced to your NADPH. 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 This is your non-cyclic phosphorylation. Now the electrons are sent here, means at photosystem 2 there is less electrons, means it is oxidized. Loss of electrons is in oxidized here. So do we need to make sure that electrons are sent there again? Yes sir, we need to send some electrons there again. What happens here? Listen to me carefully. In your inner membrane thylakoid, in the inner thylakoid membrane, all of you remember thylakoid structure? Remember thylakoid structure? In the inner membrane of your thylakoid, photolysis happens here. What happens here? I'll write this inside. One second students, let me erase this. Photosystem 2, I'll write that side. Photosystem 2, I'll write this side. Photosystem 2. So what is happening here? To make sure that electrons are balanced, photolysis is happening here. What is the meaning of photolysis? Your H2O. Remember I told you in the starting, water, plants need water for survival. And the hydrogen source is actually the water here. The water here is splitting up. It is splitting up into 2H plus 2H plus oxygen plus 2 electrons. Now these 2 electrons are sent here. This oxygen is sent to air, that is as a byproduct. Now what happens to this 2H plus? They are sent in ATP formation. They are sent to ATP formation. Now that reminds me, where is ATP formation happening here? Is there any ATP formation? Yes sir, there is ATP formation here. ADP is getting converted to ATP, adenosine triphosphate. That's why it's called as phosphorylation, addition of phosphate. And we also have NADPH formation. Clear? Clear? Is the concept clear to all of you? This is photolysis in the inner thylakoid membrane. I'll make sure I'll write some points to you so you can remember. Want to take a screenshot? Did you write down? Did you write down? All of you write down? Did all of you write down this concept? Clear? Non-cyclic is clear? What is happening in non-cyclic? In non-cyclic, first we have photosystem 2, which works at 680 nanometer. Light is coming, electrons are excited, electrons are taken by your primary electron acceptor, that is your pheophytin. From pheophytin, electrons goes to your PQ. What is PQ? Elastoquinone. Do you want me to write all this information? So do you want me to write what is PQ? PQ is plastoquinone. Plastoquinone. Okay. Plastoquinone. What is CY? CY is nothing but your cytochrome B6F complex. Cytochrome B6F complex, it's small b, okay? Small b complex. What is PC? PC is nothing but your plastocyanin. Plastocyanin. And what is F? FD? FD is ferredoxin. Now, one last thing to mention here. Is which enzyme is involved. Here, this conversion. In this conversion, we have an enzyme called as FNR enzyme. FNR enzyme. Now, this FNR enzyme is called as ferredoxin NADPH. Wait, there's no space here. Ferredoxin NADPH reductase. NADP reductase. That is the enzyme which is involved. This happens in the outer thylakoid membrane. This happens in the inner thylakoid membrane. It is photolysis. Photolysis. Clear, all of you? Clear? Is this concept clear to all of you? Non-cyclic photophosphorylation. Now I'll make sure I will write some points about this concept. You also should write down, and I'll tell you why are they important, because questions will come. Statement based question will come from here. Okay, statement based question might come from here. Okay, so write down the first point here. The first point I want all of you to write down here is, in non-cyclic phosphorylation, O2 is evolved. Yes sir, O2, where's the pen here, sorry. In non-cyclic phosphorylation, the O2 was evolved here. Yes, O2 was evolved. Second point can I write? Photolysis happening here. That is splitting of water. Splitting of water in the inner thylakoid membrane. In the inner thylakoid membrane. The third point I want to focus is ATP synthesis happening here. Yes sir, ATP synthesis is happening here. The fourth point here is NADPH is produced. NADPH is produced. Where is it produced? In the outer membrane. In the outer membrane. The fifth point I want all of you to focus here is the electron which was sent here. Is it coming back to your photosystem 2? The electron which was received by your photosystem 1, which is used here, is it coming back here? No sir, it is not coming back. So can I call it non-cyclic? The next point is non-cyclic. It is non-cyclic. And the last point is, does this look like a Z? Does this look like a Z? Yes sir, it looks like a Z. So can I call it Z scheme? Yes sir, I can call this Z scheme. The sixth point here is, it is also called as Z scheme. It is also called as what? Z scheme. Yes, both NADPH as well as ATP are synthesized. Okay, done? Non-cyclic, any doubt? Non-cyclic, any doubt here? You should tell me right now. I will clarify the doubt for non-cyclic. First tell me. Any doubt? Easy? All of you know it is very easy now, right? Non-cyclic is very easy. All of you wrote down with me. All of you wrote down these points. What happens? Because students, question will come from these statements also. Question can come from, I'll tell you where question can come from. I will tell you where the question can actually come from. Aren't these handwritten notes? These are handwritten notes, right? So this is, question can come from here. Question can also come from the sequence. Pheophytin, PQ, CY, PC, PC to your FES, FES to FD. Now the sequence is there, right? This sequence also can be asked as a NEET PYQ. It has been asked. So please remember the sequence as well as the events. The events and the sequence as well as the electron acceptors, the final electron acceptors, the photolysis is happening. That's all. Any question from non-cyclic photophosphorylation you should be able to do by now. Okay? In the PDF I'll send. In the PDF I'll send. Clear? And in non-cyclic photophosphorylation, both your PS2 is involved as well as PS1 is involved. Electrons from PS2 is transferred to your PS1. Electrons from PS2 is transferred to PS1. Clear? Amazing. All of you are actually learning, I believe. I can see some spam in the chat, but I'm going to ignore that. But I'm so happy that all of you are actually willing to sit for this long and learn these concepts. Okay? Now, can we go to the next concept? All of you are willing to learn. I can see that all of you are willing to learn all these concepts. So that shows hunger. Okay, can we start with cyclic now? Can we start with cyclic? Cyclic is even more easy. Cyclic is even more bacha, easy concept is, easy peasy lemon squeezy it is. Can we start? Can we start with the cyclic phosphorylation? Now question for all of you. Question for all of you is that the cyclic, okay, this is a mic. Question for all of you. When does the cyclic phosphorylation happens and where does it happen? Simple question. Simple question is, when does it happen and where does it happen? The answer is very simple. Cyclic happens whenever the wavelength more than 700 nanometer is used. Whenever wavelength more than 700 nanometer wavelength is, whenever there is wavelength of light more than 700 nanometer, cyclic photophosphorylation happens. Yes sir, correct. Now the cyclic photophosphorylation, can this happens in the thylakoid membrane? In your thylakoid we have your P680, that is your PS2. Then we also have PS1, that is 700. Can cyclic happen here? Can cyclic happen in your PS2? No sir, cyclic cannot happen here because cyclic will only happen when wavelength is more than 700 nanometer. So it only happens in PS1. It only happens in PS1, mainly in your stroma lamella. On your stroma lamella, between the two thylakoid connection, where we only have your PS1, there cyclic happens, and in this PS1 the cyclic happens. Clear that point? I hope that point is clear to you. I'll draw the diagram. Please draw with me. All of you should draw with me. So I told you we have your main reaction center. What is the meaning of main reaction center? We have your chlorophyll A, that is main reaction center. Then we have your antennas. We have multiple antennas, also called as your accessory pigments. All sunlight is coming. Only the wavelength more than 700 nanometer is captured now by your photosystem 2. By your photosystem 2. So photosystem 1, sorry, photosystem 1, which has a wavelength of your 700 nanometer. Photosystem 1. Now this photosystem 1, it's a pigment. Chlorophyll A is a pigment. On any pigment, photon is coming and blasting on it. Electrons are emitted. Yes, electron is emitted here. 2 electrons are emitted. 2 electrons are emitted. Electrons are then taken up by your primary electron acceptor. Yes sir, electrons are taken up by primary electron acceptor. What is it here but? The answer here is again same, that is FES. Your iron sulfur centers. Now from here the electrons go to your FD. Again FD. Yes sir, it goes to FD. Now from here, where does the electrons go? From here, electrons go to your plastoquinone. From here, electrons go to plastoquinone. From plastoquinone, it goes to your your cytochrome B6 complex, B6F complex. From here, it goes to your PC, that is your plastocyanin. Now from plastocyanin, it goes back. The electrons go back to the reaction center. They go back to the reaction center, that is your chlorophyll A. And in this process, is NADH being produced? The answer is no. In your cyclic photophosphorylation, there is no NADH produced, absolutely absent. But is ATP being produced? Yes, sir. But in this, is ATP being produced? Yes, ATP is being produced here. ADP is getting converted to ATP. Here also, ADP is getting converted to ATP. ADP is getting converted to ATP here. Only and only ATP is produced. And where this is happening mostly? This reaction is mostly happening in the stroma lamella. Why? Because of the photosystem 1. Any doubt here? Any doubt here? Ask me right now in your cyclic photophosphorylation. Clear? Concept clear? Marks with you? Concept is clear? Marks is with you? Yes, sir. You made the concept clear and marks is also with you. Telegram channel. My Telegram channel is simple. You just type my name. What is my? Basavaraj. I think Basavaraj Sir. Just type this. My Telegram channel will come. Clear? No doubt. Amazing. Let's go to the next concept. I'm so thrilled now because next is again very easy. Oh, next is a question. Next is a question. Are you all of you ready to do some questions? Is each and every one of you ready to do a question? Is everyone ready to do questions? Tell me. If I see fire, if I see lighting, if I see, yes, if the light is exactly 700 nanometer, if it's more than 680 at 700, it is taken up by your photosystem 1. Yes, next I'll be teaching respiration. That's again very easy chapter. Respiration is even more easy. Respiration is even more easy. Next I'll take respiration. Next I will take respiration. Let's do some questions. This is a NEET PYQ last year. Fast, answer this question. Easy. The reaction center of PS2 has an absorption maxima. PS2 will only observe where? This reaction will happen. This reaction will happen only when the wavelength is more than 680. Whenever wavelength is more than 680, right? PS2 cannot take it. Your PS2 cannot take it. So it goes to PS1. Only PS1 will be active when the wavelength is more. Okay? NEET PYQ, done and done. 4 marks, all of you. Write down. Plus 4 marks for all of you. Such an easy call. I don't know why all of you are struggling with this chapter. Very easy chapter. This is a NEET PYQ 2023. The next question. Use the Vibgyor, which I taught you. Students, use the Vibgyor concept and tell me the answer for this question right here. First, use the trick. Use the trick first. Use the trick and show me the answer. Use the trick and show me the answer here first. Take your time. Use the trick. Vibgyor. First. Fast. Let me sit. My legs are hurting a little. Use the trick and do this answer. You will have plus 4 marks. This is also a NEET PYQ, students. I'm not using random questions. I am showing you the proof that whatever I teach in my class is useful for you in your NEET preparation. I mostly teach based on the questions. Okay? Done? The answer is first, that is A is 3, that is A is blue green, yes, Vibgyor blue green, then B is 2, yeah, 1, it is 1, B is 2, then xanthophyll is 4, then keratinoids is yellow to orange. Next question fast, answer. I want to ask this question. Which of the following statement is incorrect? Which of the following statement is incorrect? Grana lamella. Grana lamella have both PS1 and PS2. Cyclic photophosphorylation involves both PS1 and PS2. Involves both PS and PS2. Both ATP and NADPH are synthesized during the non-cyclic photophosphorylation. Yes. Stroma lamella. Stroma lamella lacks PS2 and NAP reductase. All of you know what is this? You know grana lamella, right? All of you know what is grana lamella? The grana thylakoids, right? What is the answer first? The answer is second one. Obviously, all of us know in your cyclic, we just learned cyclic photophosphorylation does not have your PS2. It only has PS1. Clear? Easy questions? Yes? Next question. Next question. The product of light reaction. The product of light reaction. Right? The product of light reaction is what? The product of light reaction in photosynthesis is what? We know in your non-cyclic, when we're doing, someone is asking why not one? Grana lamella. See, remember, this is your grana lamella. The thylakoid membranes, grana. This is your grana, right? Which will have both your PS2 and PS1, both will have, right? It will have both. Who can tell me the answer for this one? The product of light reaction in your non-cyclic photophosphorylation, we learned that ATP is produced, NADH is produced, and also we know when the water is splitting up, your what oxygen is released? Oxygen is released. Option number A. Did anyone say anything else? Did anyone tell this was option B? Because water is not a product. Water is actually splitting up. Clear all of you? Water is splitting up there. Now, can we start the next concept? Are all of you able to apply the concepts which you learned today in today's class in questions? So students, after today's class, just go read NCERT once. Just go read NCERT once. It's going to be so easy for you. Okay? It is not 3. It is not 3 because hydrogen is not, H2O is not evolved. H2O is splitting of water is happening. H2O splitting of water is happening. Okay? Can we start the next concept? That is chemiosmotic hypothesis. The most easiest concept. The most easiest. Most easiest. Most easy. Most easiest concept. Can we start the next concept? Amazing, all of you are here. So, forget this definition, I'll tell you. Forget the definition, I'll tell you what is chemiosmotic hypothesis. Chemiosmotic hypothesis tells you how ATP is produced inside the chloroplast. That's all. Sir, is there someone is asking me, sir, won't there be any situation when only PS2 is working? No. PS2 is involved in non-cyclic. Remember, it is involved in non-cyclic means both PS1 and PS2 both should be involved. Both should be involved there. Okay. Chemiosmotic hypothesis tells you that it is a hypothesis which is telling you that, what is it telling you? How ATP is made inside the chloroplast. Clear? Clear? How ATP is produced inside the chloroplast. Are you with me? Are you with me, all of you? Chemiosmotic hypothesis says that how is ATP produced inside the chloroplast? That's all. That's all. But now I'll tell you how it happens. Okay? I want full Josh now. I want full Josh because I'll be teaching you one small concept which will help you in the next chapter also. I'll teach you a small concept which will help you in the next chapter also. That is nothing but your how are ATP synthesized. Ready with me? Can you start? Can we start? Fast, tell me. I want Josh. I want energy from all of you. If all of you agree, I'll teach you now. I'll teach you how energy is obtained. Okay. Now full Josh, I will tell you how are ATP synthesized. Now in total there are 3 methods. All of you know in biology there are 3 methods by which ATP is produced. What is the first method? Can anyone tell me what is the first method which happens in the glycolysis? The first method which happens in glycolysis, which is called as substrate level phosphorylation, right? Substrate level phosphorylation. That's the first method, where directly glucose is converted to pyruvate. Using a substrate. Yes, that is substrate level. This happens in glycolysis. All of us know this. If you do not know, don't worry. I will teach this in the next chapter. Okay. The next one which is, someone has a doubt? Sir, more than 700 means PS will take place, guys. And see, in case of your cyclic, right? In the case of cyclic, I said more than 680, actually not 700, more than 680 nanometer. Harsh, in case of cyclic, whenever there is wavelength of light more than 680 nanometer, that time, more than 680, that time photosystem 2 cannot work. Only and only photosystem 1 will work for that time. Okay. The second one is called as your photophosphorylation. Photophosphorylation. What is the meaning of photophosphorylation? With the help of light energy, we are producing ATP. Did we just learn that in cyclic and non-cyclic? In cyclic and non-cyclic, a wave, light was coming, photons were coming, exciting the electrons, and ATP was produced. That is the second type which we just learned. And the third type is your oxidative phosphorylation. Oxidative. This is extra information which I want all of. This is information which is given in your NCERT actually, but this is going to be useful for your next chapter also. Oxidative phosphorylation. What is the meaning of oxidative phosphorylation? You are using oxygen to make ATP. You are using oxygen to make ATP as an electron carrier. This happens in the mitochondria. This happens in the mitochondria. Clear till now? This three are enough. I will teach you now this part. I will teach you this now. Is this clear? Is the diagram clear to all of you? I see very less likes. I don't know why. I see so many less likes. I don't know. All of you like the video right now, and then I will teach you Chemiosmotic hypothesis. Okay. Now listen to this very carefully. Open your NCERT if this is not clear. If this is not clear, please open your NCERT right now with you. Open your NCERT. Keep it next to you. Okay. Keep it next to you. NCERT. Okay. So I told you in chemiosmotic hypothesis. ATP is produced. Yes, sir. In chemiosmotic hypothesis, ATP is produced. Yes. But how is it produced? That is the answer I'm going to tell you now. Okay. Now this is your thylakoid membrane. Now this is, if there is photosystem 2 and photosystem 1, both are here. Yes sir, both are here. If both are present here, is this cyclic or non-cyclic? All of you should tell me. Is this cyclic or non-cyclic? Tell me fast. This is non-cyclic. Yes sir, this is non-cyclic. Light is coming, falling on this. Electrons are getting ejected. Electrons are getting ejected to your plastoquinone. I told you. From pheophytin to plastoquinone. From plastoquinone to your cytochrome BC oxidase. Then to your PC. That is your plastoquinone. So cytochrome B6F complex. Here everything. Plastocyanin PC. Students, till now in your NCERT they never mention these words. When they are teaching you cyclic and non-cyclic, they have never used word. But look at here. Look here what NCERT is doing to you. Look what NCERT is doing to you. Everything it is mentioning here. And then it is going to PS1. From PS1, in the outer membrane, in the presence of FT, FNR, that is ferredoxin, NADP, reductase, NAD is getting into reduced to NADPH. Yes sir. Now listen to me very carefully now. Listen to me very carefully how ATP is produced. That's what the theory is. Listen to me very carefully. To produce the ATP, right, we need 4 requirements. How many requirements, students? To make this entire thing work, it is 4 requirements are needed. What are the 4 requirements? Where will I write it? Where will I write it? I don't have space to write, students. Where will I write? Okay, I'll draw a separate here. To make the entire thing work, there are 4 requirements. The first requirement here is to make the ATP, we need 4 requirements. The first requirement is H plus gradient. There needs to be a H plus gradient. Yes, first point. To make the ATP, we also need a intact membrane. Yes, we need a intact membrane. If you open your NCERT, you will find these points. If you open your NCERT right now, these points are mentioned there, but it is not telling you why it is required. Okay, to make the ATP, you also need a proton pump. You need a proton pump to pump the protons. And the last point, the last requirement is nothing but your ATP synthase. What do you need? You need ATP synthase. ATP synthase, that is nothing but your F0 F1 complex. F1 F0 F1 complex, which is a transmembrane protein. It is a type of transmembrane protein. These are the 4 requirements. Write down, students. Write down this. I will erase this right now. I will erase this because I have to go to the next slide, because this is not a proper slide. I will erase this. Please write down this. Please write down this one. To make the ATP, we need to make the chemiosmotic theory work, we need a H plus gradient should be maintained. A gradient should form. We need an intact membrane, that is double membrane. Then we need a proton pump to pump the proton. The last requirement is a ATP synthase. We need a something called as a transmembrane protein called as ATP synthase. Done? All of you done? Can we go to the next Blackletter diagram? Now, the first point was what? To form a gradient. Now, students, what is the meaning of gradient? Gradient means a difference. We form a difference here. We make a difference here. Of what? Of H plus ions. Of H plus ions where? In the lumen. So students, please remember, this is your thylakoid membrane. Remember, I drew the thylakoids like this. Thylakoids. This is a one of them. This is a zoomed version of it. Zoomed version. This inside part is there, right? Inside part is this. And the inside part is called as what? Lumen. The inside part is called as what? Lumen. Now inside the lumen, we need to increase. Increase what? H plus ions. Means what? We have to make lumen acidic in nature. Can I call it acidic? Yes, sir. We need to make the lumen acidic in nature. Lumen has to be acidic in nature. So how do we do it? How do we make the difference? How do we make the difference? How do we make the H plus gradient? The first point all of you should focus here is this. We know when H2O is photolysis happening. Whenever the photolysis is happening, we got the oxygen which is emitted out. We got H plus. We also got electrons. Right? We also got electrons. Electrons were taken up. Electrons went away. Right? Electrons were excited. They went away. And this H plus, I told you when I was when I was drawing the non-cyclic, I told you that H plus is involved in ATP synthesis. Did I tell you not? Did I mention that time? Did I mention that this H plus is involved in ATP synthesis? Yes. So we are getting H plus here. So H plus concentration is increasing. Yes, it is increasing. Now, next one I want all of you to focus here is, the next point I want all of you to focus is here. Whenever the electron is being transported here, whenever the electron is getting transported here to your plastoquinone here, this part, see, they have written specially, everything is mentioned here. This plastoquinone will act as a hydrogen acceptor. Will act as a what? A hydrogen acceptor. Okay, listen to me very carefully now. This will be act as a PQ will act as a primary hydrogen acceptor. It will accept the electrons, and this PQ has become PQH. Will become PQH. Can you see here? It's written in NCERT also. NCERT also says here PQ is getting converted to PQH. Now when the electron is sent to the next, when the electron is sent forward to your cytochrome B6, the H plus is sent to your lumen. The H plus is sent to your lumen. And once again, once the H plus is sent to the lumen, this becomes PQ again. This becomes PQ again, and H plus is coming from your hydrogen acceptor. Can I tell H plus concentration is again increasing? Yes, sir. H plus ion concentration is increasing inside the lumen. Yes. Now, next point I want all of you to focus. Yes, plastoquinol. It becomes plastoquinol. Yes, PQH. The next point I want all of you to focus here is here. NAD plus is taking up a proton and it is becoming NADPH. It is becoming NADPH. Where it is happening? In the outer membrane, outer thylakoid membrane. So can I tell here H plus concentration is decreasing? Yes, sir. H plus concentration is decreasing here. H plus concentration is increasing. First from here, second from here. So here it is increasing, here it is decreasing. Do you form a difference? Do you see a difference here? Do you find a difference? Do you find a gradient here? So gradient formation done. First step done. Clear all of you? Is everyone with me here till now? Is everyone with me till now? I want everyone's answer till now here. Have you understood how we form the proton gradient? Yes, in the stroma, H plus is decreasing. In the lumen of your thylakoid, the H plus is increasing. Yes, you are with me, all of you? Now, listen to me very carefully now. Now, this H plus goes to your, this is your F naught, this is your F naught, this is F1, this is F naught, this is F1. Whenever the protons pass through this, whenever the protons are passing through this, what is happening? There is a small motor there. There is a small motor there. The motor is turning. Whenever the motor is turning, can you see this part? The outer part here. which is outside the thallic membrane, this part is turning. Whenever it turns, there is a conformational changes are happening, and when this is turning, right, ATP formation is happening. What is happening? ATP formation happening from ADP to ATP formation. This is nothing but your ATP synthesis through chemiosmosis. ATP synthesis using chemiosmosis. We need a proton gradient, we need an intact membrane, we need a motor pump, and we need a ATP synthase. And we need a ATP synthase here. Clear? Did anyone have a doubt here? Does anyone have a doubt here? Does anyone have a doubt how this works? So as protons are moving, this is rotating. Once this rotates, ADP is getting converted to ATP. ADP is getting converted to ATP. Clear, all of you? Understanding? Amazing. This is chemiosmotic theory. This is nothing but your chemiosmotic theory. Clear? Awesome. All of you are understanding. That's amazing. You are making my job easy. You are making my job easy. Did everyone understand? So here, proton gradient one from here, one from here. Here it is decreasing, increasing, going through. No doubt. In my notes you will find this. In my notes you will find this. What I just mentioned, right? How is the proton gradient happening? Splitting of water. That thallic membrane, that hydrogen is coming inside. And the third one is your NADPH. In my notes you will find this. Whatever I just explained to you is written here. It is also written to you in your NCERT. Harsh is saying, I have a doubt. Please ask your doubt. Please ask your doubt. Please ask your doubt. Yes, yes, ask your doubt. It's fine. Ask your doubt. I'll answer it. Therefore, if no one has a doubt, can I ask a question? Can we solve a question? Can we solve a question, everyone? I have a question now. Can we solve a question? Fast, tell me. Can we solve a question? I believe none of you have doubt. All of you know what is cyclic, what is non-cyclic, what is chemiosmotic theory. Everything is clear till now? Done. So please explain cytochrome B6X one more time. Sir, the increase in concentration of hydrogen goes where, sir? So please explain cytochrome. See here, students. What exactly happens in your cytochrome B6 here is nothing but before B6, before cytochrome B6, there is something called as PQ. Yes, if you remember the non-cyclic, in non-cyclic I told you the first electron acceptor. The first electron acceptor is your plastoquinone. No, where is it? The first electron acceptor is your pH. That is, first electron acceptor in your non-cyclic is pH, right? The first electron is pH. Is your pheophytin. Pheophytin is accepting the electron. Now from pheophytin, the electron is going to your PQ. From PQ, the electron will go to your CY. That is, CY is nothing but your cytochrome B6F complex. From CY it goes to your PC, that is your plastocyanin. Then from here it goes to your PS1. But when it is getting when the electron is transferred from your pheophytin to your PQ, whenever the electron is transferred from your pheophytin to PQ, the PQ, that is plastoquinone, gets converted to PQH. That PQH is nothing but your plastoquinone. Plastoquinone. So PQ to PQH, and then again it gets converted to PQ. This is a cycle. This is a cycle which is happening here. But whenever the cycle is happening, right, this H plus is sent. This H plus is sent to the lumen. To the lumen. Clear? Sir, if H plus concentration increases, then what happens? If H plus concentration is increasing, when the H plus concentration is increasing, it is making a potential gradient. Whenever you have a difference, students, whenever there is a difference now, here more H plus, here less H plus. So H plus will always travel from higher concentration to lower concentration. Yes, from higher concentration, it is going to lower concentration. So it is passing through here. When it is passing through here, this motor is rotating. It is rotating. When it is rotating, right, ATP is synthesized. Sir, impermeable to membrane, then how it comes out? Because this particular ATP synthase is a transmembrane protein, Harsh. It is a transmembrane protein. This protein is present between the membrane here. That is why only and only here it can come out. That's why it cannot come out here anywhere. Your H plus cannot go anywhere outside here. Only and here because we have a transmembrane protein. It is a protein which is present between your membrane. Clear? Clear, all of you? Can we solve a question? Doubts are amazing, students. You take up the doubts. You might not have some of students will be clear, but as soon as someone asks a doubt, right, they'll be like, oh, how does it happen? So students, doubts are extremely important. Okay, doubts are extremely important. Can we go to the question? Can we go to a question now? Here's the question. This is a question from your NEET 2023 PYQ. 2023 last year PYQ. This is a last year PYQ. Please solve this question. Question to you. Question to all of you. Yes, alpha. Not exactly in the stroma. It happens in your outer membrane of thylakoid. It happens on the outer membrane of thylakoid. Answer this question. So students, if you were to write the NEET 2023 paper last year, this was the question given to you. Easy question. Can everyone in the chat tell me? Everyone should type in the chat. Sir, it is a very easy question. Very easy question. Very easy question. Which of the following is requirement for chemiosmosis, that is ATP production? We need a membrane, intact membrane, proton pump. We need a proton gradient, that is H plus gradient. H plus is proton. And then we need ATP synthase. Yes, all of you get 4 marks. All of my students who are learning from me today will get a 4 marks if this question comes again. So students, every topic I'm telling you, every topic I've teach you till now is question oriented. I am teaching you concept based on questions. Okay? Now, what have we done till now? We finished light reaction, light dependent reaction, cyclic is done, non-cyclic is done. So can we start dark reaction now? Can we start dark reaction? Can we start with dark reaction, all of you? In the chat to tell me. We are coming to end of the chapter actually. We are coming to end of the chapter. After dark reaction, you only have factors affecting photosynthesis. I will, Ravindrani, I will solve more questions. I will make sure bring more questions to you. Do one thing. See, time is limited on YouTube. What I'll do is, I will post questions today only. I will post, no, tomorrow, today's late again. I will post questions on my Telegram channel tomorrow morning. Will all of you solve questions tomorrow? I will solve questions. I will add questions tomorrow. All of you know my Telegram, right? Basavaraj Sir. I know my name is old fashioned, but I am not. So just go to my Telegram channel. Tomorrow morning I will add so many PYQs. Okay? Dark reaction. Now, in the starting of the chapter, in the start of today's chapter, I told you, in the starting of today's chapter, there is something called as PCR cycle. I will take on YouTube also. I'll take a complete session on PYQs. I'll take. Don't worry. In the starting of the chapter, I told you something called as PCR cycle. Right? PCR cycle. What is PCR cycle? PCR cycle is nothing but PCR cycle is nothing but your photosynthetic carbon reduction cycle. So your carbon is getting fixed here. That is nothing but your dark reaction, also called as light independent reaction. This is also called as your C3 cycle. It is also called as your C3 cycle. Okay? It is also called as your C3 cycle. Can we start? Can we start? Photosynthetic carbon reductase cycle. So students, in your light reaction, you learned that ATP and NADH is produced. Yes sir, in your light reaction, ATP and NADH is produced. And that ATP, that NADH is used in your dark reaction. It is also called as your biosynthetic phase. Why is it called as biosynthetic? Because in your dark reaction, starting 5 minutes of class I told you, in the 5 minutes of class I told you, CO2 is getting converted to C6H12O6. CO2 is getting converted to C6H12O6. That is called as biosynthetic phase. Because in the dark reaction, we are fixing the carbon dioxide and forming the products. We are making sugar. In your dark reaction. And what are we doing here? It requires the products of your light reaction. In your dark reaction, it is craving for your NADPH and ATP from your light reaction to synthesize sugar. To make sugar, we are utilizing the light reaction products. I told you, photosynthetic carbon reduction, PCR cycle. Carbon from the atmosphere is fixed to form sugar, already discussed. The reaction requires enzymes present in the stroma. So where does it happen? This reaction, the dark reaction, happens in the stroma of chloroplast. Point is clear, sir? Point is clear? That is stroma of the chloroplast, dark reaction is happening. Yes, sir. So, what does it require? What are the requirements? I told you already. The raw materials are your ATP, NADH and CO2. The raw materials for your dark reaction to happen is ATP requirement, NADH requirement and CO2 is also required to make the dark reaction. So CO2 enters the cell. The CO2 is entering the cell through stomata. That is why your photosynthesis is called as what? Physico-chemical process, because there is exchange of gases here. Physico-chemical process. Physico-chemical process. So before I teach you, no, this I'll teach you later, but yes, I will teach you all the enzymes. Wait, have patience. I'm building up the hype. I am hyping up your C3 cycle. In your dark cycle, the first CO2 fixation. I told you the raw material for your dark cycles are nothing but your ATP, NADH and CO2. The CO2 is getting converted, is getting fixed in different type of plants. We have C3 plants and we also have C4 plants. We have C3 plants and C4 plants. Now this dark reaction is a little different here. In C3 plants, listen to very carefully, students. Listen very carefully. In C3 plants, the first stable CO2 fixation. In C3 plants, the first time when the CO2 is getting fixed, you get a 3 carbon compound. We get a 3 carbon organic acid called as PGA. You get a PGA. You get PGA in the case of C3 plants. That is a 3-carbon compound called as phosphoglyceric acid. Called as phosphoglyceric acid. In C3 plants, CO2 fixation is happening. In C3 plants, when the CO2 gets fixed, we get a 3-carbon compound called as phosphoglyceric acid. But in the case of C4 plants, the first stable CO2 fixation, when CO2 is getting fixed, in C4 plants, a 4-carbon organic acid is produced called as OAA, also called as oxaloacetic acid. Now I will be using these two when I draw the cycle. I want all of you to identify which is the first stable product in C3 plants, the first stable product in C4 plant. In the case of C3 plants, the first stable product is nothing but phosphoglyceric acid, which is a 3-carbon compound. That is why they are called as C3 plants. But in the case of your C4 plants, the first stable organic acid in case of C4, the first stable is nothing but your 4-carbon oxaloacetic acid. Okay? Now, can we do C3 cycle? I'm just building up the hype. I will draw it right now. So please draw with me. In your C3 cycle, we mainly have 3 steps. The first step is carboxylation. Second step is reduction. And the third step is regeneration. The C3 cycle is also called as your Calvin cycle. Why is it called as Calvin cycle? The scientist called as Melvin Calvin. The scientist known as Melvin Calvin, during the World War II, in your Second World War, during the Second World War, he used radioactive isotopes on an algae. And he first time deduced the steps for your C3 cycle. Or C3 cycle. Okay? C3 cycle. Now write down with me, all of you. Please write down with me. In C3 cycle, the first acceptor. In C3 cycle, the first acceptor of carbon dioxide. The first acceptor of carbon dioxide is none other than RUBP. What is RUBP? Can anyone tell me what is RUBP? RUBP is nothing but rubulose 1,5 bisphosphate. What is RUBP? RUBP is nothing but rubulose 1,5 bisphosphate. Yes sir, all of you know what is RUBP. Whenever RUBP comes in contact with your CO2. Now this RUBP is a 5-carbon compound. 5-carbon compound. Here 1 carbon is coming. 1 carbon plus 5 carbon. How many? 5 plus 1 is 6 carbon. But do we get a 6-carbon compound here? No sir. We get a 2 molecules of 3-carbon compound. 2 into PGA. This PGA is a 3-carbon compound. PGA is a 3-carbon compound. What is PGA? PGA is nothing but your phosphoglyceric acid. 3-phosphoglyceric acid. Now, this first step is catalyzed by an enzyme. Can anyone in the chat tell me what is the enzyme here? The most abundant enzyme. Which is the enzyme? First tell me, who can tell me the enzyme here? The enzyme which is present here is none other than your Rubisco. What does CO means? Rubisco, CO means carboxylase as well as oxygenase. And this first step This first step here is called as carboxylation. Carboxylation. You are fixing the carbon here. You are fixing the atmospheric carbon. Now what happens? Now this particular PGA is getting converted to, one molecule of PGA is getting converted to triose phosphate. What is it converted to? Triose phosphate. Triose phosphate. Now this triose phosphate, when the conversion is happening, this is again 3 carbon, triose means 3. When this conversion is happening, this step here is called as reduction. So I told you here, if you see, first step was decarboxylation, CO2 fixation. Second step was what? Reduction. In the second step, 2 ATP. Write down with me. In the second step, 2 ATP gets consumed to ADP. In the first step, 2 ATP is getting converted to 2 ADP. Along with that, your NADP DPH is getting converted to, reduced to NAD+. So in your reduction, in your second step, 2 ATP is getting consumed. Also 1 NADH is getting completely consumed. Now from here, we go back to your RUBP. Right? We go back to RUBP. Now this step, the third step, is called as regeneration. Is called as what? Regeneration. In this regeneration step also, ADP is getting converted to ATP. Sorry, ulta. What am I writing? Has my brain already gone dead? In this also, ATP is being consumed to ADP. ATP is getting consumed to ATP. No, but sir, you told me in C3 plants, but where is the sugar? Where is the product? The product is here. This triose phosphate later gets converted to sucrose, and later it is stored as starch. Sir, how many ATPs and how many ADHs are formed? Okay, Harsh is asking a simple doubt. Harsh is asking me innocent doubt. Sir, how many ATP and NADH are formed here? The answer to that question is 0. Mote. 0. There is no formation. There is no formation of your ATP, no formation of NADPH here. Only and only consumption is happening. I'll tell you a trick here. 3210. 3210. What is the 3 here? The 3 here is how many ATPs are getting consumed here? The ATP getting consumed here are 2 plus 1. 3 ATPs are getting consumed. Right? 3 ATPs are getting consumed. How many NADH are getting consumed here? This is actually 2. Because each 1 PGA is there, right? 2 PGA. So 2 NADH is getting consumed here. NADPH is getting consumed. How much of CO2 is getting is used? 1 CO2 is getting used here. How much of ATP formation? How much of energy generation is happening here? 0. 0 energy happening here. 0 energy generation is happening here. Clear? Is this concept clear to all of you? Is this concept clear? Oh, my legs are hurting a little. Is this concept clear to all of you? Where is the pen? NCERT telling you a diagram, no, there is no consumption. There is no consumption at all. Complete zero energy is made here. Only energy is used. Only energy is used here. Okay? How many ATP? 3 ATPs are used. 2 NADH. 1 CO2 is getting fixed. And zero energy. Okay? This is NCERT diagram. What is this RUBP? RUBP, rubulose 1,5 bisphosphate. Atmospheric CO2 is coming, and as well as hydrogen. What is carboxylation? It is getting fixed. It's getting fixed. We have your PGA. How many PGA? 2 PGA. Here, 2 PGA is formed here. 2 PGA. This is your 3 carbon compound. 3 carbon compound. This is your 5 carbon compound. Okay, 5 carbon. Now PGA to triose phosphate reduction is happening. ATP and NADH are used up. Sucrose and starch is made. From triose to regeneration is happening. Again, ATP is consumed. And finally we get regeneration of RUBP. Yes, I know Kannada because I am from Karnataka. I am from Bangalore. I've been growing up here all my life. I've been growing here all my life. Bangalore. Clear, all of you? Any doubt here? The next concept I want all of you to learn here is, for every CO2 molecule, I told you, for every CO2 molecule, please listen to this. This is a NEET PYQ. Resonance is asking, what is PG? Sir, how many NADP is produced? Students, doubts, show me the doubts. So what is PGA? PGA is nothing but your phosphoglyceric acid here. PGA is your 3-phosphoglycerate. 3-phosphoglycerate is your PGL. Then someone is asking me, how many ATP? There is no ATP or no NADH production. Complete zero. Ravindrani, look at this trick I told you. 3, 2, 1, 0. Zero energy production. Zero energy production. There is no energy used here. No energy produced. Only energy is consumed. Okay? Only energy is consumed here. Energy is not released here. Clear on that point? Is everyone clear on that point? Energy is only consumed here. ATP is getting consumed. 3 ATP. 2 NADH is consumed. And 1 CO2 is also consumed. Energy releasing, no energy releasing. Okay? How many NADP is consumed? 2. 2 here. 2 NAD. 2. 2 NADP is consumed. Clear? Now, here. For every CO2 molecule entering, all of you know, I showed you CO2 is entering the chat. Like all of you enter the chat right now. CO2 is entering the cycle. Yes, CO2 is entering the cycle. For every CO2 get fixed, for every CO2 to get fixed completely, we need 3 molecules of ATP. I told you 3 molecules of ATP, and Ravindrani 2 molecules of NADPH are required. For every CO2 fixation in one cycle, we need 3 molecules of ATP and 2 molecules of NADH. Clear? Hence, energy required to fix 6. Energy required to make 6 CO2. Why 6 here? Because we know in the balance equation there's going to be 6. So if they can ask you in examination, if they will ask you in examination to fix let's say 8 CO2, they can ask you in examination, students, instead of 6 they can ask you 8 CO2. Here I have given you for 6. This is also given you NCERT. For 6 CO2 we need 18 ATP. Why, sir? Why do you say 18 ATP? Because for 1 we need how many? For 1 we need 3. So 3 into 6. 18 ATP. 6 into 2, 12. 6 into 2, it is 12. So students, in the examination, if a question comes this year in NEET examination, how many ATP and how many NADH, how many ATP and how many NADH is required to fix 8 molecules of CO2, what is going to be the answer? Yes, 6 cycles makes up 1 glucose because 6 carbon are in your glucose. C6H12O6. C6H12O6. So 6 carbons are required. Yes. In your NEET examination, if a question comes this time, we have 8 to fix 8 CO2. How many ATP and how many NADH we need? All you need to do is 6 into 8. 6 into 8 for ATP. So what am I doing? 8 into 3. 8 into 3. 8 into 3 here. And we need to do 8 into 2. We get this. Okay? 8 into 2 and 8 into 6. But here they have given you 6 here because when you use 6 CO2, 1 glucose is made. Very important line. Star statement. When 6 CO2 is consumed, 1 glucose is made. All of you are amazing. You're able to answer. Okay? Correct. 16 NADPH, 24 ATPs. Clear? Now these are the examples of your C3 plants. It could be your wheat, soybean and oats. Question, take up. NEET 2023 or 2020, I don't remember. Do the answer first. I'll just drink some water and come. I'll just drink some water and come. While I go and come, drink some water. I don't have a water bottle here. I go take a cup of water, drink and come. And then I want answer for this question. Okay? 2 minutes, students. 1 minute. Done. Easy question. Obviously, this is a NEET PYQ. All of you know the answer now. It's 18 as well as 12. How do you know? All of you know this, right? How it is formed also, all of you will know. How do we get 18 here and 12 here? From C3 cycle calculation. Because in C3 cycle, to fix 1 CO2, we need 3 ATP and 2 NADH. Amazing. Now, can we start photorespiration? Can we start with photorespiration, all of you? Now, can we start with photorespiration? All of you, everyone thumbs up in the chat. If I can start photorespiration. Now I told you, students, listen to me very carefully now. Golden mutts. Yes, these are golden mutts. Do you know this enzyme called Rubisco? This enzyme right here, Rubisco. This particular enzyme. Yes, after C2 we'll do C4. Okay. this enzyme right here called as Rubisco, this enzyme right here can attach itself. This enzyme right here, this Rubisco enzyme, ready? Can we start? Okay. C4 we'll do later. So this enzyme Rubisco right here, Rubisco enzyme, it is called as Rubisco, right? What is the meaning of C here? Carboxylase. What is the meaning of O here? Oxidase. It is this particular enzyme. This particular enzyme my brain is not braining little. This particular enzyme right here, this will help. This particular enzyme will help in making the reaction. What reaction? Here RuBP is sitting. This is the enzyme RuBP sits here in the substrate, and this here CO2 sits here. It is trapping CO2. It is trapping CO2 and it is getting converted to your PGA. Yes, it is getting converted to PGA. But this particular enzyme Rubisco, can also do oxygenase. What is the meaning of oxygenase? This particular enzyme can also trap oxygen. So instead of your CO2, instead of capturing the CO2, it is catalyzing the oxygen. That is your photophosphorylation. So in photophosphorylation, it occurs in your C3 plants. It can also trap oxygen. It happens in your C3 plants. Does the photophosphorylation happen in the case of C4 plants? Absolutely not. Please remember this concept. Photophosphorylation will never happen in the case of your C4 plants. It only happens in the case of your C3 plants. And O2 is consumed here. Yes, in the case of photophosphorylation, photorespiration, O2 is getting consumed and CO2 is released in the process. CO2 is released outside. And Rubisco oxygenate says RuBP. So RuBP plus oxygen is getting converted here. Write down the steps here. Write down the steps, all of you. Here, in the case of your photorespiration, we have RuBP. In the case of photorespiration, RuBP joins with oxygen. RuBP joins with oxygen and it converts. It makes up a 3-carbon PGA. Yes, a 3-carbon PGA. Plus, it also makes a 2-carbon molecule. Can anyone in the chat tell me what is a 2-carbon molecule? The 2-carbon molecule is phosphoglycolate. Phosphoglycolate. Now this phosphoglycolate is a 2-carbon molecule. Yes, it's a 2-carbon molecule. It is a 2-carbon molecule. So can you see here, RuBP is 5-carbon. Do we have any carbon here? No. So that's why this 5-carbon has to split up. That's why it's split into a 3-carbon here and a 2-carbon phosphoglycolate. 3 plus 2 is 5. Simple maths. 3 plus 2 is 5. Now this particular glycolate undergoes a lot of changes and it gets converted. This phosphoglycolate undergoes changes and it gets converted to your PGA. While it is getting converted to PGA, CO2 is released. CO2 is released out. CO2 is released out. Now, in this entire process of photorespiration, I want you to write down some points. Is there any sugar formation? We know in C3, triose phosphate was forming sugar. But in the case of photorespiration, also called as C2 cycle. Why is it called C2 cycle? Because the first stable product here is second carbon. In this case of photorespiration, there is no sugar formation. Yes, sir, there is no sugar formation here. I can all of you understand that. Is there any ATP formation? Usually, in the case of C3 also, there is no ATP formation. So, no ATP formation. There is release of CO2. Plants need CO2, right? Plants need CO2 to do C3. But in the case of your photorespiration, CO2 only is released outside. CO2 is released outside. In this case, ATP is getting consumed. Yes, sir, ATP is also getting consumed. It is also ATP. It is also eating up your ATP. And last point, which most important point here is, it does not happen in C4 plants. This photorespiration does not happen in your C4 plants. So, do you see any advantage to this process? Do you notice any advantage, any form of advantage to this process right here? Absolutely not. This is a most useless process which is happening in a plant, also called as wasteful process. It is a wasteful process. It is no advantage, complete wastage happening in a plant. Okay, but you can ask me now. You can ask me right now, sir, if it is a wasteful process, why is this even happening? Can anyone in the chat tell me why is this even happening? It is happening because of the environmental conditions. It is happening because of the environmental conditions. What environmental conditions? When is the photorespiration happening? This point, which I'll tell you right now. This point is going to be your NEET PYQ. Trust me. This is going to be a NEET question. When will the photorespiration happen? When will the photorespiration happen? Write down this. When will the photorespiration takes place? Simple. Why this process happen? This process is happening because of the environmental conditions. There are certain changes in the environmental conditions. Because of the changes in the environmental conditions, this process is happening in the case of C3 plants only. Only in C3 plants. Where is it happening? First case. First case is when in the environment, oxygen concentration is completely very high. When the environment, in the environment, there is so much of oxygen. When there is so much of oxygen, your photorespiration is happening. The second point, whenever there is very high light intensity in your temperate regions, all the regions which is close to the equator, very high amount of light from sun. So whenever there is high light intensity, photorespiration is happening. All this can be asked as a question. The third point, whenever there is very, very high temperatures, extremely high temperatures, photorespiration will happen. Okay. And the fourth point, whenever there is extreme low concentration of CO2. Whenever there is no CO2, very low concentration of CO2, photorespiration will happen. Clear? Write on this point. When will the photorespiration happen? Write down. Clear? Any doubt here? Any doubt here, students? Any doubt here? I can see very less likes today. I don't know why. Very less likes, very less students also today. Students are decreasing. I think you did not share the video or you don't like the video. Clear? When will the photorespiration happens? What happens in photorespiration? In photorespiration, your RUBP in presence of Rubisco oxygenase, it is adding to oxygen and your 2 carbon compound is formed, and this is again getting fixed to your PGA, but that time CO2 is released. There is no sugar, no ATP, release of CO2, consumption of ATP. It happens, does not happen in C4, only and only it happens in your C3 plants. This is called as photorespiration, the wasteful process. The wasteful process, when does it happen? When high concentration of oxygen, high light intensity, high temperatures and low concentration of oxygen. Amazing. Now, the last but second concept now. The last but second concept for today is, how did your C4 plants fight against photorespiration? How did your C3 plants, C4 plants fight against this particular type of photorespiration? They fought against it. How? They made sure whenever the fixation of carbon is happening, whenever the C3 cycle is happening, whenever the fixation is happening, there is complete absence of oxygen. There is complete absence of oxygen. Yes, complete absence of oxygen. In the case of your Kranz anatomy, which is seen in the case of your monocots, which show C4 cycle. Do you see these vascular bundles? Around the vascular bundles, we have thick bundle sheets. What do we have? We have thick, large, with no intercellular spaces. We have bundle sheets. Bundle sheet cells. And around the bundle sheet cells, what is this green color? These are your mesophyll cells. What do we have here? Mesophyll cells. So mesophyll cells are surrounding your bundle sheet cells. Bundle sheet cells surrounding your vascular bundles. This is the anatomy of your Kranz. In the case of your Kranz anatomy, that is leaf anatomy of C4 plants, that is called as Kranz anatomy. The bundle sheet cells completely surround, arranged in a wreath. It is arranged in a wreath around the vascular bundles. Around the vascular bundles, bundle sheet cells are completely arranged. And around these bundle sheets, you have your, what do we have? We have your mesophyll cells. Bundle sheet cells are characterized by your having large number of chloroplasts. Yes, bundle sheet cells are extremely large. Yes, bundle sheets are called as very large, they're very thick, and there is no intercellular space here. Completely intercellular space is absent. So can your oxygen enter inside? Can oxygen and CO2 enter inside easily? No sir. Your bundle sheet cell will completely be impervious. They'll make sure nothing is entering. That is, C4 plants are high temperature tolerant. So where does C4 plants take place? They are present in your equator region. On the equator region, C4 plants are present and they show high temperature tolerance. They respond to high light intensities. They lack photorespiration. Photorespiration is completely absent. And greater productivity of biomass, more energy is produced here. What happens here? In the case of your C4 cycle, more energy is produced. So someone in the chat was telling, C4 cycle is also called as Hatch and Slack. Hatch and Slack cycle, because the two scientists who discovered it, that is your Hatch and Slack. That is two Australian scientists called as Hatch and Slack discovered the C4 cycle. They found that in tropical plants which are growing near the equator, a tropical, very high light, high temperature, high light, that are much more efficient in CO2 utilization. They are not doing any wasteful process. They are not doing, remember, your C4 plants are not doing your photorespiration. Yes sir, they are not doing photorespiration. And the first stable product, very important from now here, I told you before also, the first stable product in the case of your C4 plant is oxaloacetic acid, also called as OAA, which is a 4-carbon compound. Clear? Till here any doubt? Till here just the basics is over, introduction is over. Can we start the cycle? All of you? Can we start the cycle? I know all of you are also getting mentally exhausted now. All of you also reached your saturation point. But students, trust me, I hope till now, whatever you have done till now, from past almost 3 hours, 7:30, 9:30, almost 3.5 hours, 3 hours 45 minutes, everything is clear to you. CAM pathway, I'll teach you separately. CAM pathway is not right now here. I will teach you Crassulus, that is Crassulus pathway that happens in different plants. Don't worry about Gautam, don't worry about your CAM pathway. I will teach you separately. How many hours still? Last 30 minutes. It's over. Okay? How can you message personally? Harsha, Telegram channel. You can message me on Telegram. I will reply to you there. Shiprasad saying is in full energy mode. Okay, let's bring on full energy. So students, I want energy in the chat now. For Shiprasad, I want extreme high energy in the chat. For 8 CO2, that was the question I asked you. Just, okay, take a 3 minutes break and we'll start this. In C4 cycle, what happens? I will teach you in C4 cycle. Take a 3 minutes break. Last, we'll have a last stretch. We'll do a last stretch. It is now your 10:22. At 10:15 we'll start. At 10:15 we'll start with full energy. 10:15. to give me 30 minutes, I'll finish it. Okay? 10:15 we'll start. Okay? while everyone joins, let me start drawing. Can we start? Students, last stretch. We'll be doing what is C4. Then we'll be doing a comparison between your C4 and C3 cycle. Then last topic we'll be doing after this is factors affecting photosynthesis. Can we start? Hello Navina. Welcome to the class. Students, if you understood everything till now, all the concepts you're doing from past 4 hours, like the video, subscribe to the channel, and don't forget to share the video, because this is a complete package of your photosynthesis in higher plants. Let's start. So all of you please draw with me. All of you please draw with me. So in the case of your C4, plants, there are two types of cells. I told you in the case of C4 plants there are two types of cells. One is called as your bundle sheet cell. Other one is called as your mesophyll cell. So let us draw the cell. Let us draw two cells here. This is called as your mesophyll cell. Then we have your we have two cells here. Alright, that side. That's fine. It's called as mesophyll cell. Upper one is called as a mesophyll cell. Lower one is called as your bundle sheet cell. Now, listen to my words very carefully now. Listen to my words very carefully. This is a very beautiful cycle. Step by step we'll write. Step by step we will write here. CO2 is entering into your mesophyll cell. But can CO2 enter your bundle sheet cell? Can CO2 and oxygen enter inside your bundle sheet? No sir, because bundle sheet cells are extremely thick, no interstellar spaces, so hence nothing can enter bundle sheet. But your mesophyll cells, yes sir, inside the mesophyll cells your CO2 is entering. Now in the case of your C3 plants, CO2 was interacting with RUBP, but here in the case of C4 plants, CO2 is interacting with PEP, phosphoenol pyruvate. What is PEP? PEP is nothing but phosphoenol pyruvate. If you remember glycolysis, even in the glycolysis in the last but one step we mentioned about PEP, phosphoenol pyruvate, right? Phosphoenol pyruvate. Now this phosphoenol pyruvate is a 3-carbon compound, right? Phosphoenol pyruvate is a 3-carbon compound. When CO2 gets fixed in the presence of an enzyme called as PEP carboxylase, carboxylation is happening. Yes, in the presence of PEP carboxylase. What are we getting? 3 carbon plus 1 carbon. We get a 4-carbon compound called as oxaloacetic acid, OAA, which is a 4-carbon compound. Yes, we get 4-carbon compound. This is getting converted to your malic acid. Whenever this is getting converted to malic acid, remember students, whenever it is getting converted to your malic acid, your 1 NADPH is converted to your NAD plus. One NADH is converted to NAD plus. Yes. All of you following with me, right? All of you follow with me, please write down. This malic acid is also 4 carbon. Now, this malic acid is sent. This malic acid is sent to your bundle sheets now. This malic acid is sent to your bundle sheet. Yes, it is sent to bundle sheet. Now here the malic acid, gets again converted to your OAA. Again gets converted to your OAA, and this OAA gets converted to pyruvate. Gets converted to pyruvate. Now when this is 4 carbon, this is 3 carbon. So 1 carbon is CO2 is released. Yes sir, 1 CO2 is released. Yes. Now where does this CO2 go? This CO2 enters again a C3 cycle here. Again a C3 cycle. This CO2 enters here. Again we have what? Again inside your bundle sheet C3 cycle is happening. If C3 cycle is happening, what needs to be here? Again we need to have your RUBP here plus CO2. We are getting your, again RUBP is getting covered here. RUBP is taking up, and the C2 C3 cycle is happening here. C3 cycle is happening here. We are getting PGA. PGA is again getting converted. So the entire process happening is C3. Now here this pyruvate is sent back. This pyruvate is sent back to your mesophyll cells. This pyruvate is sent back to your mesophyll cells. That is pyruvic acid or pyruvate. Pyruvic acid, 3 carbon. It is sent back here. Now from this pyruvic acid, again this is converted back to your PP. From your pyruvate, it is getting converted back to your PEP, and while doing so, 2 ADP is getting converted to, 2 ADP, sorry. While doing so, your 2 ATP is consumed. While doing so, 2 ATP is getting consumed to your 2 ADP. Now what happens is NADH? What happens is NADH? In this process here NADH, NAD plus, this NAD plus was also transferred here. NAD plus is again transferred to your, converted to NADPH. NADPH. This is the entire C4 cycle. Clear? Clear all of you? I'll repeat once again. In the case of your C4 cycle, we have mesophyll cells, we have bundle sheet cells. 2 separate cells are there. In the case of mesophyll cells, CO2 is entering inside. CO2 is reacting with your phosphoenol pyruvate or phosphoenol pyruvate in the presence of PEP carboxylase. What it is, with the help of PEP carboxylase, it gets converted to your OAA. What is OAA? Oxaloacetic acid. Now this oxaloacetic acid is 4 carbon. How? 3 carbon plus 1 carbon, 4 carbon. Oxaloacetic acid is getting converted to your malic acid, 4 carbon. When this is happening, NAD plus, NADPH is getting converted to your NADP plus. Malic acid is transported to your bundle sheets. Yes, it is transported to your bundle sheets. In the bundle sheet, it is getting converted to OA again 4 carbon. Now this OA is getting converted to pyruvic acid, which is 3 carbon, means a carbon dioxide is lost. This carbon dioxide enters your C3 cycle again. So in the case of C4 cycles, C3 cycle is also happening in the bundle sheet, and also in the bundle sheet RUBP is present. If RUBP is present, Rubisco is also present. Yes sir, even Rubisco is also present here. But here in the case of mesophyll cells, we have PEP carboxylase. Now pyruvic acid is sent back here. Once pyruvic acid is sent back here, we need to do regeneration of pyruvic acid to your pepcarboxylase. When the conversion is happening, 2 ATP is consumed. 2 ATP is consumed. That is your C4 cycle. Jyoti is asking me, please state about what NEET deleted syllabus. Jyoti, your wish is granted. I will make a separate video this week itself or next week. Next week I'll make a video on botany chapter wise, what you should study, what you should not study. Okay? Okay, Jyoti, just for you, I will make a small video on telling that which topic is in botany is in syllabus, which is out of syllabus, which topic you should study from your new NCERT, which topic you should study from your old NCERT. Okay? Don't worry, I'll do that for you. Clear? Any doubt here, students? Any doubt here? Crystal clear? Amazing. All of you are amazing. Now, can anyone tell me in C3 cycle how many ATP and how many NADH was consumed? Fast. Someone tell me in the chat in your C3 cycle, how many ATP and NADH were consumed? In the case of your C3 cycle, if we remember correctly, 3 ATP and 2 NADPH was consumed. Right? NADPH was consumed. In the case of your C3 cycle, I'm talking about in general. I'm not talking about 6 glucose. I'm not talking about 6 carbon, 6 CO2. I am talking about to fix 1 CO2. To fix 1 CO2, 3 ATP plus 2 NADPH was consumed. Clear? But in the case of C4 cycle, C3 is also happening. So can I write 3 ATP plus 2 NADPH plus the 2 ATP here, plus 2 ATP here. Students, this right here is a good comparison between C3 and C4 cycle. The energy consumption, the amount of energy consumption between C3 cycle and C4 cycle. Trust me, this can be asked as a question. How much amount of energy is consumed in C4 cycle? You should tell me 5 ATP plus 2 NADPH in C4 cycle. Clear, all of you? See here, students. This here NADPH is not getting consumed. It is just getting transferred here. It is not getting consumed. It is getting transferred here. That's all. Okay? Clear? All of you clear with this concept? All of you are amazing. All of you can easily write NEET examination. I know for a fact. Can we do a quick comparative study before we go to the last topic for today, which is going to be a maximum of 20 minutes topic. Last topic is 20 minutes. Before we go to the last topic, can we do a comparative analysis of entire mesophyll bundle sheet? Also C3, C4. Can we do? Yes sir, we can clearly do. In mesophyll cells, carry out light dependent and independent reaction. Yes, in the case of mesophyll cells, both are happening. Mesophyll cells, because we know in the case of C3 plants, we have thylakoid membrane, everything is happening. But in bundle sheets, majority is carried out light independent. Mostly dark reactions only and only happens in the case of bundle sheet. Only and only in the case of bundle sheet, majoritively dark reaction takes place. It is very thin walled, mesophyll is very thin walled, but your bundle sheet is thick walled. Why? Because it has to be impervious to the oxygen, right? Absence of starch granules are absent here. Presence of starch granules. CO2 acceptor here is PEP. Right? PEP will accept the carboxylase in mesophyll in C4, and here CO2 acceptor is RUBP. That is C3 cycle, the common C3 cycle, which is happening inside the bundle sheet. C3 cycle is happening. So RUBP takes up the CO2. Here, RUBP takes up the CO2. Clear? Can we do one more comparative now? This is a table which is given in your NCERT. Table which is given in your NCERT. What is the first question here? Cell type in which Calvin cycle takes place. Where does the C3 cycle takes place? Asking you, where does C3 cycle take place? In the case of C3 plants, mesophyll cells, because only one cell is there. So C3 cell Calvin cycle. But in the case of your C4 plants, bundle sheet cells. Clear? So after each point I tell you, just say clear sir. So I will know that you are understanding. Okay? Can I get that? So after each question of each point, I will tell you if it is clear. You should tell me if it is clear or not. Okay. Now, cell type in which initial carboxylation reaction. Initial carboxylation where is happening? In the case of C3 plants, it's also mesophyll cells. Even in the case of your C4 plants, mesophyll cells. Initial fixation. C4 plants, again mesophyll cells. Done. Clear? How many cell types does the leaf have that fixes CO2? In the case of C3, only one. In the case of C4, we have two types. That is mesophyll cells as well as bundle sheet cells. Clear? Amazing. Which is the primary CO2 acceptor? Primary CO2 acceptor. In the case of C3 plants, it is RUBP. Yes, RUBP is taking the CO2. But in the case of your C4 plants, it is PEP carboxylase. PEP, phosphoenol pyruvate, takes up the initial CO2 fixation. Number of carbons in the primary CO2 acceptor, it is 5 here. That is RUBP has 5 carbon, while your PEP carboxylase has 3 carbon initial. Right? CO2 acceptor. Is this point clear? Is this point clear, all of you? Is this point clear? Last point here. Number of carbons in the primary acceptor, that is RUBP 5, PEP carboxylase only and only 3. The 3 plus 1, that is CO2, it becomes OAA, which is 4 carbon. Clear? Next one. Which is the primary CO2 fixing product? What is the CO2 fixing? In the case of C3 plants, PGA is obtained. First product, first stable product, PGA. In the case of your C4 plants, we just assured you, it is oxaloacetate, the first stable product. The first stable product is OAA. Number of carbons in the primary CO2 fixation product, PGA is 3 carbon, while your OAA is 4 carbon. Does the plant have rubisco? Yes, both of them will have rubisco. In the case of your C3 plants, rubisco is present in the mesophyll cells, but in the case of your C4 plants, rubisco is present in the bundle sheet. Remember that. Does the plant have PEP carboxylase? Absolutely not, because PEP carboxylase, the first fixation is not happening. In the case of C4 plants, yes, PEP case is present. Which cell the plant have rubisco? I told you already. Mesophyll cells. Here it is bundle sheet cells. In the bundle sheet cells only that your C3 cycle will take place. Clear point? CO2 fixation rate under high light condition. Whenever there is high light condition, will the CO2 fixation happen? Here it will be medium to low. Why low? Because we know whenever there is high light condition in CO2, photorespiration takes place. High light condition, photorespiration can take place. So your medium work will happen. Very medium work will happen. Here if it is obviously high, because your C4 plants are amazing. They can work in your high light condition. Whether photorespiration is present at low light intensities, we know at low light, whenever there is low light, photorespiration will not happen, because I told you the conditions for photorespiration is high light, high temperature. So low light, it will not happen. And obviously in C4 plants, photorespiration does not happen at all. Clear this point? Is this point clear, all of you? Are all of you following with me, or I'm talking to myself? Are all of you following till now, or all of you following me, or am I talking to myself? Have I gone insane? Students, then whether the photorespiration is present at high temperatures, at higher temperature, obviously photorespiration will happen. Yes, I told you, in higher temperatures photorespiration is happening. Here obviously photorespiration will not happen because in C4 plants photorespiration is absent. Whether photorespiration would be present at low CO2 concentration, photorespiration at low CO2, when CO2 is so low, obviously oxygen will be taken up. Yes sir, photorespiration will happen. I told you the condition I told you. Here obviously it's not happening. Whether photorespiration would be present at high CO2, absolutely not. Whenever there is amazing CO2, there is high CO2 is present, obviously the plant will do C3 cycle. It will not do your photorespiration. So negligible. Optimum temperature 20 to 25 degrees Celsius. High temperature 30 to 35 degrees Celsius. Example is wheat, rice. Example is maize, sugarcane and sorghum. Maize, sugarcane and sorghum. If all those concepts are clear to you, solve this question. Solve this question first. Solve this question. Given below are two statements. The primary CO2 acceptor in C4 plants. The primary electron acceptor, carbon acceptor. The primary CO2 acceptor in C4 plants is phosphoenol pyruvate, PEP, and is found in your mesophyll cells. It's found only in mesophyll cells. Mesophyll cells in C4 plants lack Rubisco enzyme. They lack Rubisco enzyme. What is the answer? This is a NEET PYQ from 2023. Think an answer, all of you. Think an answer. That is, obviously it is correct because we know PEP takes up the CO2 and gets converted to OAA. Yes, CO2 plus PEP gets converted to OAA. And we know that in the case of mesophyll cells, where only fixating is happening, that is malic acid is forming, your Rubisco is absent, only PEP carboxyl is present. But in the case of your bundle sheet, in bundle sheet, it is present. Can we start the last topic? 10-15 minutes and I will finish the chapter. We have gone from the most basic to the advanced level, students. Most basic to the advanced level today. Most basic to the advanced level. I hopefully all of you are following what I'm saying. Hopefully you're understanding this concept and hopefully this chapter is becoming easier to all of you. Can we start? The last topic for today is factor affecting photosynthesis. Factor affecting the photosynthesis. Now, listen to me very carefully now. Listen to me very, very carefully. Factors affecting photosynthesis, there are two factors. One is called as internal factors, one is called as external factors. The internal factors are based on the plant's genotype. The internal factors depend on your genotypic predisposition. This is an NCERT line. This is an NCERT line. So what are the internal factors? Internal factors such as the number, size and age and orientation of the leaf. If the leaf is like this or if the leaf is like this. If it is flat, more sunlight. If it is like this, less sunlight. Number of the leaves, more number of the leaves, more photosynthesis. Large size of the leaf, more photosynthesis. Young leaf, more chlorophyll, more photosynthesis. Number of mesophyll cells, more photosynthesis. Number of chloroplast, yes, number of chloroplast is very important. And amount of chlorophyll. All of these are internal factors which plant has on itself based on its genetic composition. Based on its genetic composition, a plant's internal factors play a role in the rate of photosynthesis and amount of photosynthate. Okay. Now, there is something called as limiting factor. Listen to me very carefully now. Limiting factor. What is limiting factor? How do I say this? Limiting factor, I'll tell you, listen to me very carefully. Limiting factor is a factor. I told you there are many factors here. There are many factors here responsible for the photosynthesis, right? There are multiple factors here responsible for photosynthesis. The factor which is least responsible, I repeat again, the factor which is the least responsible is called as limiting factor. It's called as what? It is called as limiting factor. I'll write here. Please write down with me. This definition is important. Many factors are involved here, right? Many factors control the biochemical reaction here. Many factors control the biochemical reaction. Yes, what biochemical reaction? The photosynthesis. Many factors control the photosynthesis. Then photosynthesis rate. The rate of photosynthesis is determined by that factor which is present in the least, which is present in the least amount, which is present in the least amount. So there are many factors. There are many factors responsible for photosynthesis, but the factor which is present in the least amount, that is called as your limiting factor. In your chemistry you also study, right? Limiting reagent. In your chemistry you study limiting reagent. Similarly, we have limiting factor. Now let me give you an example. Let me give a quick example. In winter mornings, in winter, early morning winter, sunlight is less. Yes sir, in early winter, in your winter season, in the winter season, early morning, light is less. So can I call light as a limiting factor there? Yes sir, light is a limiting factor there because light is present in the least amount there. But as the day goes on, as you have afternoon now, we have enough of light, right? We have enough of light now. Then in the winter season, what about then? Light no more remains the limiting factor. Light no more remains the limiting factor. Now I'll give you one more example. In your summer, in the case of your summer, initially when the light is just coming out, that time light is a limiting factor. Yes sir, light is initially a limiting factor. But again, as summer, we know summer gets hot. In summer, it gets hot. So we have enough of light. As the afternoon approaches, we have enough of light. That time is the light limiting factor? No sir. But remember, in summer, there is less water. Yes sir, in summer, there is less water. So that time, water becomes the limiting factor. So whichever factor is present in the least amount, I repeat again, whichever factor is present in the least amount is called as rate determining, also called as limiting factor. Clear all of you? Is the concept clear? Is the concept clear? Limiting reagent is clear to all of you? Amazing. So, cool, all of you know. Now, clear all of you? Limiting reagent. Now let's go to external factors. Let's go to the external factors. The first one is your light. How light plays a crucial role. How light affects the photosynthesis. The first you need to know here is in terms of light. The first thing you need to know in terms of light here is light. There are many types. Light, there are something called as light intensity. What is light intensity? The amount of light which is falling. Right? Light intensity is nothing but your concentration of light. Right? Concentration of light. So can I call in the afternoon regions, in the afternoons? In the afternoons, light intensity increases. Yes, sir. In the afternoons, light intensity increases. That is the light intensity. Yes. Then we also have something called as light amount. Then we have something called as light amount. The amount of light is coming there. The amount of light. For example, some of the plants need large amount of light. For example, we have something called as long day plants. They need a lot of light. Then we also have short day plants which need less light. That is the amount of light. Second point is the light. Then we also have something called as quality of light. We know light is not very important. We need blue light and red light. That is more important. That is the quality of light. Right? That's the quality of light. That's the quality of light. But which factor out of these three? Which factor out of these three, that is amount, intensity and quality, which plays a crucial role in photosynthesis? The answer is intensity. The answer is intensity. Intensity controls. Intensity controls photosynthesis. This is the point which all of you should remember. Out of the three things, amount of light, intensity of light and quality of light, out of these three factors, the intensity of light controls the photosynthesis. Is the point clear? Is that point clear to all of you? Is that point clear to all of you? Now, this is a graph which is given in your syllabus. This is a graph which is given in your syllabus. Now, if you look here, they have graphed light intensity to the rate of photosynthesis. As the light intensity is increasing, here, the rate of photosynthesis is also increasing. Yes, sir. All of you can say this. All of you can agree with me. As the light intensity is increasing, the rate of photosynthesis is also exponentially increasing. Yes. So this statement is correct. Intensity of controls the photosynthesis. Yes, Shreyas sir is here. Yes, sir. It's been 4.5 hours. Last 10-15 minutes I'll end the class. Last 10-15 minutes I'll end the class. There are a lot of students here from the starting. Okay. Now, from this particular graph, can I tell this OA? This particular starting point is O. This particular pointing point is O. And till here it is A. This particular region is OA region. In the OA region, can I tell rate is directly proportionate? In this region, rate is directly proportionate to intensity of light. Right? Rate is directly proportionate to the intensity of light. As the light is increasing, as the intensity of light is increasing, rate is also increasing. Yes, sir, it is directly proportionate. But if you look at A to B, from here, B to C, from B to C, in your NCERT it is given A to B or A to B in your NCERT. In this region, do you see a saturation? Yes, sir, we see a saturation there. What is the meaning of that? The meaning is very simple. At this point, right, at this point, how much hour of intensity you increase. How much of intensity you increase, the photosynthesis will be constant. What is the meaning? From this point onwards, light is no more a limiting reagent. It is no more a limiting factor here. Can I say at this point rate is constant? Rate is constant from here. Rate becomes very constant, that is, it reaches a saturation. It reaches a saturation here. Reaches a saturation here. Clear all of you? Clear all of you? Is this point clear till now? How light intensity plays a role in the rate of photosynthesis. In the rate of photosynthesis. So this can show you one thing. This can mean one thing. Light is actually very rarely a limiting reagent. Usually, whenever there is light intensity, photosynthesis increases. So here NCERT says a line. NCERT mentions one line that light intensity is rarely a limiting reagent. Is rarely a limiting factor. Rarely a limiting factor. It is never, only in the case of your shaded plants, like your money plants. All of you have money plants in the house. Only in the case of few cases such as your money plants, it can become a limiting factor. But in the other plants, usually light intensity is rarely a limiting factor. But you should know how light intensity plays a role for the photosynthesis. Clear? Is that point clear to all of you? Is that point clear to all of you? Just say yes, sir, I'll go to the next one. Just say yes and I'll go to the next one. And do you know this saturation is reached just at 10% of sunlight. Just at 10% of sunlight, the saturation can be reached. Just at 10%. Just at 10% saturation, this is will reach. Now one more, NCERT says one more line. NCERT says one more line, something called as high intensity. High intensity. If there is high intensity light, right? If there is very high intensity, your rate of photosynthesis decreases. If high intensity photosynthesis decreases because the pigments can get damaged. Clear? If it reaches more intensity, the pigments gets damaged. Clear? See, alpha, you are confusing. You're talking about nanometer. Nanometer comes under quality of light. This is the intensity of light. There is a difference. Intensity of light is different than quality of light. Quality of light, where you read nanometer. This is intensity of light. Okay. Is in light clear? Is this clear? Can I go to the next concept? This is where we talk about your wavelength, that is blue and red. Quality, duration also is important. The next one is carbon dioxide concentration. And this carbon dioxide concentration is the most important. Why? Because it is a major limiting factor in the photosynthesis. This is a NEET PYQ. They'll ask you directly, which is the most limiting factor? It should be carbon dioxide. Why? Why is carbon dioxide limiting factor? Because in your environment, carbon dioxide is so less. Just 0.038%. Just 0.0% of carbon dioxide is present in your atmosphere. It is just roughly around your 380 parts per million ppm. Around just 380 parts per million, right? In the environment, it is so less. That is why it is a limiting factor. But what you need to understand here is, for C3 and C4, how it is different. That's where the question will come. How your carbon dioxide plays a role for C3 as well as C4. Now listen to me very carefully. Graph is important here. Graph is important. Listen to me very carefully. In the case of your low light, write down with me all of you. In the case of low light, whenever there is less light, right? Light is limiting factor now. In the case of low light intensity, now when there is low light intensity, this is a limiting factor, and very high carbon dioxide, very high carbon dioxide. Will there be any effect? Will be there any effect in photosynthesis? No, sir. No effect. Because your limiting factor is here CO2. There will be no change in the photosynthesis in your C3 and C4 because low light is the limiting factor. But in high intensity, in high light intensity, whenever there is very high light intensity, who will benefit? Tell me fast. Whenever there is high light intensity, who will benefit? Whenever there is high light intensity, your C3 plants will benefit. Who will benefit? C3 plants. Why, sir? Why are you telling me C3 plants? Because the saturation point for carbon, how much amount of carbon someone can take, how much amount of your, no, don't get me wrong, Lakshmita, I'll tell you, that is a different concept. Are telling me C4, that is different separately. I'll tell you why it is C3. Because I didn't tell you one point, my bad, my bad. I didn't tell you this. The saturation point, it means how much a plant can absorb the CO2. How much a plant can absorb CO2, that saturation point for your C3 plants. The saturation point for your C3 plants is around your 450 ppm. Around 450 ppm. But for your C4 plants, it is just around your 360 ppm. Around just 360 ppm. Just around 360 ppm. Just around 360 ppm. Now you tell me if there is very high intensity of light is available in the nature. So is light a limiting factor? No, light is not a limiting factor now anymore. And we also have high carbon dioxide. Which plant will benefit more? If there is more carbon dioxide in the environment, obviously C3 plants will take more CO2. They will convert more CO2 fixation, more energy liberation. Yes, more energy liberation. What about C2 plants? C2 plants can just take around 360 ppm, almost 360 to 365 ppm of CO2. Will they benefit much? No, they will not benefit much. That's why in your greenhouses, in the greenhouse, do you know what is greenhouse? In your greenhouse, we completely cover it so that whenever the light is coming inside, light is trapped inside. If you walk inside the greenhouse, you can see warmth. There is so much of hot temperature inside. Inside your greenhouse, there is very high temperature and also there is very high CO2. Very high CO2 in your greenhouses. And which plant grows in greenhouse? In your greenhouse, we grow C3 plants. We grow C3 plants in your greenhouse. Because C3 plants has higher capacity of carbon absorption. And hence, they grow, they are able to produce more energy. So how does carbon dioxide play a role here? Carbon dioxide will play a very important role in your photosynthesis because in the case of your C3 plants, more photosynthesis is happening. Because saturation point of your C3 plants is very high. But in the case of C4, it is little less. Clear? Is that concept clear to all of you? Why in your greenhouse? In your greenhouse, we grow C3 plants such as your bell pepper, capsicum, the red color capsicum, green color capsicum, and also we grow tomatoes, Because those plants, the C3 plants, has a higher capacity to absorb the carbon dioxide. Clear? All of you? Clear? Amazing. All of you are amazing. This was just a graph here. This was just a graph showing that C4 plants can absorb more, C3 plants are less. Which is higher? This is more. C4. Clear? Next one. Temperature. Last two. Stick with me. Last two we'll do and then you're free to go. Can we do the last two one? Last one is temperature. This is water. Now students, you tell me. In high temperatures, do enzymes work? Tell me, all of you tell me. In high temperatures, do enzymes work? No. Enzymes doesn't work in high temperatures. Enzymes gets completely denatured. Enzymes gets completely denatured in the case of high temperature. Yes. What about less temperature? What about if the temperature is very less? If the temperature is very less, enzymes again becomes inactive. In less temperature, enzymes become inactive. So remember that in the case of temperature, we need an optimum temperature for photosynthesis. Now let's compare in dark and light reaction. Your temperature will affect both. Temperature will affect both light and dark reaction. But remember this one point here. It will affect more. It will affect more dark reactions. Dark reaction is more affected because in case of dark reaction, there are more enzymes. Remember, in dark reaction, there are more enzymes are involved, such as your rubisco, such as your pep carboxylase, right? All the enzymes are involved here. So more enzymes are involved here. More enzymes are involved. More enzymes are involved here. Clear, all of you? Is the enzyme temperature part clear? Is the temperature part clear to all of you? Clear? Affect dark reaction more, because in dark reaction we have more enzymes are involved. Is that point clear, all of you? Now I'll ask you a simple question. In light reaction, which enzyme can anyone name an enzyme? Can anyone name one enzyme in light reaction? In light reaction, do you remember FNR? In light reaction, you remember FNR? Ferredoxin NADPH reductase, which is converting NADP to NADPH. Yes, this will also get affected if there is high temperature or low temperature. Clear? The last one is water. Remember students, when it comes to water, it directly does not affect. In case of your water, it does not affect directly. In fact, the water affects indirectly. Sir, how does it affect indirectly? How does it affect indirectly? The answer is very simple. Listen to me. In the first case, we have a leaf. We have a leaf here. If there is less water, the leaf will start to curl. Yes, whenever there is less water, the leaf will start to curl, or the surface area will decrease. Decrease in surface area. If the leaf surface area is decreasing, is the photosynthesis increasing or decreasing? Photosynthesis is decreasing. Decrease in photosynthesis. Yes sir, photosynthesis is decreasing now. First point. The second point here is very simple. Whenever there is less water, can the stomata remain turgid? No sir. If the stomatal opening, if the stomata has to be open, We need more water inside it. Yes, it has to be turgid. If there is no turgid, if there is no water, the stomatal opening will close. Yes, if there is less water, the stomata will close. Stomata will close. If the stomata is closing, you tell me everyone, if the stomata is closing, will be there exchange of CO2? Tell me first. Will be there exchange of CO2? No sir, CO2 is not entering. CO2 is not entering now. If CO2 is not entering, will the dark reaction happen? No sir, dark reaction will not happen, and hence photosynthesis will decrease. And hence photosynthesis will decrease. Photosynthesis will decrease. That is water. That is water. Clear to all of you? So what we did your light reactions, dark reaction, Kelvin cycle. We had 3 steps: carboxylation, reduction, regeneration. C4 cycle we did initial fixation, transportation, final fixation and regeneration. After that we did the factors affecting. First we did your light, then carbon dioxide, temperature and water. Temperature and water. So students, I think that's the end of the chapter. Yes, that is the end of the chapter. Let me bring it here. So that was the end of the chapter today. Complete photosynthesis has been done and dusted. So students, I want each and every one of you, everyone who has attended from the very starting with me till now, everyone should like the video. First of all, subscribe to the channel, and also please comment down any doubts you have. Please comment down any doubt you have. If you do not have any doubt, please let me know how the session was. That's what I've been asking from all of you. Please let me know how the session was. If you enjoyed the session, if you're able to understand today's session. And finally, can you tell me in the chat today, photosynthesis higher plants has been done and dusted. So comment down students, photosynthesis in plants done and dusted, and that's it. That's it. Here. And I want all of you to, here, PYQs from this chapter. Yes, I want every one of you to attend the DPP. That is VDPP. Questions are there in the description. Go on, do the description. Go on and solve the questions. Solve the questions are here. Join the Telegram channel. Every single notes will be provided to you. Questions I will be giving on my Telegram channel. You can join my Telegram channel also. And do not forget tomorrow. Do not forget tomorrow we have. If you go to live section, tomorrow we have our resonance and structure. So go on, like the video right now, and you must like the video. So you already liked here. Like the video, and I'll see you in the next class, all of you. So take care, all of you, and I'll see you in my next class, where I'll be doing the next chapter of photosynthesis. That is nothing but respiration in plants. That is going to be even more interesting, because I'll be teaching you glycolysis, Krebs cycle, electron transport chain, every single thing. So take care students, I'll see you in the next class. Please comment down, let me know how the class was today, and take care. Good night all of you. Bye bye.